Hello there and welcome to Life Beyond Seen, where the deep past moves quietly through the dark like something enormous just below the surface of still water, patient and unhurried, waiting to be noticed. Tonight, we are not going anywhere on a map you would recognise. We are not travelling to a mountain range or a coastline that exists today. We're going somewhere much further than that, not in distance, but in time. We're going back to a world so different from our own that if you could somehow stand at the edge of it, you would not recognise the sky, the smell of the air, the sound of the water, or the creatures moving through it. We're going back roughly 400 million years, give or take a few tens of millions, to a warm, shallow sea that once covered the land we now call Central Europe. A sea that lapped over what would eventually become Germany, Belgium, and the surrounding regions. A sea with no name because there was no one alive to name it. There were no birds calling overhead, no rustling of leaves from any tree you would recognise, no large animals moving along the shore. The land nearby was low and strange, covered in primitive mosses, early vascular plants, and spreading mats of green that had only recently learned how to grow above the waterline. The air was warm and thick, and the coastline was quiet in a way that is almost impossible to imagine now. Not peaceful quiet, not the quiet of a forest at midnight, but something older and emptier than that. The quiet of a world still in the process of becoming. And in the water, beneath the surface of that warm, shallow, brackish sea, something moved, something large, something that had no reason to fear anything in the water around it, because there was almost nothing in the water that could threaten it. Something that had existed in one form or another for millions of years before this moment, and would continue to exist for millions of years after. Something that, if you had somehow found yourself swimming in that sea, would have been the last thing you ever saw. We do not have a name for this creature from any ancient language, because no human was there to speak one, but we have given it a name since. A long, careful, scientific name that carries within it the memory of the man who first studied its relatives and the region of Germany where the evidence of its existence was eventually found. We will come to that name in a moment. For now, I just want you to sit with the image of that sea. Warm water, soft sediment on the floor, algal mats drifting gently, early fish moving in cautious schools near the surface, and somewhere below them, resting or moving slowly, a shape that dwarfs everything else in the water. Something that would have been, by any reasonable measure, the largest arthropod that ever lived on this planet. But before we go any further into that ancient water, I want to ask you something simple. Where are you right now as you listen to this? Are you in bed with the lights already off, the room settling around you into its nighttime quiet? Are you somewhere cool, or is it warm where you are? Is it late, or have you stolen a little time in the middle of the afternoon to let your mind wander somewhere it does not usually go? I ask, because tonight's story is one of those stories that feels different depending on where you are when you hear it. If you're near any body of water, even a river, even a pond, it might feel a little closer than you expect. And if you're nowhere near water at all, well, that might be for the best. If this is the kind of quiet, unhurried storytelling that you enjoy, I would be genuinely grateful if you took a moment to like this video and subscribe to Life Beyond Seen. It is a small thing that makes a real difference in helping this channel reach other people who might need a calm place to think and listen. There is no pressure, of course. But if this feels like your kind of evening, I would love for you to stay. Now, let us talk about what we actually know. The creature we are here to explore tonight is called Jaekelopterus renaniae. It is not a dinosaur, it is not a fish, it is not anything that most people have a clear picture of when they first hear its name. It belongs to a group called the Eurypterids, which we will explore in much more detail shortly, and it holds a record that has stood, so far as we know, since the day it was first announced to the scientific community in 2007. It is the largest arthropod ever discovered, larger than any crab, any lobster, any spider, any centipede, any insect that has ever lived. Larger, in fact, than most adult human beings standing upright. an animal built from segments and shell and claws, moving through an ancient sea with a calm, unhurried authority that comes from being at the top of a food web that had no real challenges. The name Jaekelopterus is worth pausing on for a moment. The first part, Jaekel, comes from Otto Jaekel, a German paleontologist who worked in the late 19th and early 20th centuries and contributed significantly to the study of early vertebrates and invertebrates. The second part, Pterus, comes from the Greek word for wing or fin, a common suffix in the names of eurypterids that references the paddle-like swimming appendages that many of them use to move through the water, and Rhenania refers to Rhenania, the Latin name for the Rhineland region of Germany, where the fossil evidence for this creature was found. So even the name itself is a kind of quiet story. A German scientist, an ancient sea, a region of Europe that has been inhabited and studied and named and renamed across centuries, and a creature that predates all of it by an almost incomprehensible span of time. 400 million years. It is the kind of number that sounds large but does not fully land until you try to feel it rather than just say it. Modern humans, our species as we currently understand it, have existed for somewhere between 200,000 and 300,000 years. The Roman Empire, which feels ancient to most of us, ended roughly 1500 years ago. The pyramids at Giza were built around 4,500 years ago. Agriculture as a widespread human practice began perhaps 10,000 to 12,000 years ago. And all of that, every dynasty, every invention, every migration and language and city and war and discovery that human beings have ever produced, fits inside a sliver of time so thin it would be almost invisible if you laid it against the full span of this creature's existence on Earth. Jaekelopterus and its relatives were already ancient by the time the first recognizable fish were learning to develop jaws. That is the world we are stepping into tonight. Not a world of kings and explorers, not even a world of mammals or reptiles or flowering plants. A world so far removed from our own experience that the only way to reach it is through the patient, careful work of people who spend their lives reading stone. Paleontologists, working with hammers and brushes and measuring tools and a great deal of quiet determination, looking at fragments of rock and asking the right questions slowly enough to hear the answers. And it was exactly that kind of patient, careful work that eventually gave us Jaekelopterus rhenaniae. Not a complete skeleton unearthed from a perfect cliff face, not a dramatic discovery in a remote wilderness. Something much smaller, and in its own way much more extraordinary, a single piece. A single fragment of an animal that lived and hunted and moved through warm water 400 million years ago, preserved by accident in a layer of Devonian-era rock in the Rhineland region of Germany, waiting quietly for someone to find it and understand what it meant. That fragment, and the story of what scientists did with it, is where we are going next. Because understanding how a single piece of fossil evidence can reveal the size, the shape, and the nature of an entire animal is one of the more quietly remarkable things that science is able to do. And in this particular case, what that fragment revealed was something that surprised even the researchers who found it. Not because it was impossible, but because it was so much larger than anyone had expected. The creature that left it behind was not a myth, not an exaggeration, not a reconstruction built from imagination and guesswork. It was real. It moved through a real sea. It hunted real prey. It grew to a real size that we can now estimate with reasonable confidence, even from a single piece of evidence, and that size, as we're about to discover, was genuinely extraordinary. The year was 2007. Not a particularly dramatic year for most fields of science, but for paleontology, and specifically for the study of ancient arthropods, it carried a quiet announcement that would ripple through the scientific community in a way that few fossil discoveries managed to do. The paper was published in the journal Biological Letters, a publication of the Royal Society, and its lead author was a German paleontologist named Markus Poschmann. The title was precise, as scientific titles tend to be. and the finding it described was, in the language of researchers careful not to overclaim, significant. What Poshman and his colleagues were reporting was not a complete skeleton. It was not a dramatic, full-body impression pressed into ancient stone. It was a single fossil fragment, a piece of a claw, recovered from a Devonian-era rock formation in the Rhineland-Palatinate region of Germany, near the town of Prüm. A single piece. And yet that single piece was enough to change what we thought we knew about how large a living creature with an exoskeleton could grow. The fragment itself is a chelicera, which is the technical name for the grasping appendage that Eurypterids, the group to which Jaekelopterus belongs, carried at the front of their bodies. Think of it roughly as a claw, though the anatomy is somewhat different from what we typically picture when we use that word. It is the feeding and grasping tool, the forwardmost working part of the animal, the thing it would have used to seize prey and bring it inward toward its mouth. And this particular chelicera, this single fossilized piece of one appendage from one individual animal, measured 46 centimeters in length. That is roughly 18 inches, or to put it in terms that might land a little more concretely, approximately the distance from your shoulder to your fingertips. One claw. 46 centimeters. From a creature that used it the way a hand might be used if hands were built from reinforced chitin and designed to close around living prey in a shallow prehistoric sea. When Poshman and his colleagues looked at that measurement, the next question was obvious and also somewhat complicated. If the claw is this large, how large was the rest of the animal? This is where the science becomes both elegant and, depending on how you look at it, a little humbling. Because there is no complete Jaekelopterus skeleton. There never has been, at least not yet. What exists is this fragment, and what scientists must do with a fragment is reason outward from it using what they already know. The method is called proportional scaling, and it relies on the well-documented anatomy of related species within the same broader group. In the case of Eurypterids, researchers have access to more complete specimens of related genera, animals like Pterygotus and Arcturimus, which belong to the same family, the Pterygotidae, and share a broadly similar body plan. By measuring the chelicerae of those better-known relatives and comparing them to the animal's total body lengths, scientists can establish a ratio. A claw of this size, in a body built along these proportions, suggests a total animal of approximately this length. Applying that ratio to the 46 centimeter chelicera from Prüm, Poshman and his colleagues arrived at an estimated total body length of approximately 2.5 meters, which is just over 8 feet. To be clear about what that means in human terms, the average adult man in Europe is somewhere around 1,75 m to 1,80 m tall. A creature 2.5 meters long, built low and wide and armoured, would dwarf that. It would extend well beyond a person lying flat on the ground. It would be, without any exaggeration, larger than a human being in every practical sense. Not taller, because Eurypterids were not built to stand upright in the way we are, but longer, heavier, and almost certainly more physically imposing in any environment they shared. The paper was received with a mixture of genuine excitement and careful scrutiny, which is honestly exactly how good science should receive an extraordinary claim. The excitement was understandable. A single fossil fragment had potentially revealed the largest arthropod in the history of life on Earth, a creature that outgrew not only every modern crab and spider and lobster, but every previously known fossil arthropod as well. The scrutiny was equally understandable, and it centred on a question that is worth sitting with for a moment, because it gets at something important about how paleontology actually works. The question is this: how confident can we really be in a size estimate derived from a single fragment? And the honest answer is that confidence in paleontology is always a matter of degree. It is never absolute, and any researcher who tells you otherwise is probably not doing the work carefully enough. What Poshman and his colleagues had was a well-reasoned, methodologically sound estimate based on established comparative anatomy. The proportional scaling method they used is not guesswork. It is the same approach that has been applied successfully to many other fossil animals, and it produces reliable estimates when the reference species are closely related and the body plan is consistent across the group. In the case of pterogortid eurypterids, the body plan is well documented from multiple more complete specimens, and the chelicera to body ratio appears to be reasonably consistent. So the estimate of 2.5 meters is not a wild extrapolation. It is a careful, defensible inference from real evidence. That said, it is still an inference. The fossil record does not hand us certainties, it hands us fragments, and we do the best we can with them. Some researchers have pointed out that proportional scaling may not hold perfectly at extreme sizes, that very large individuals within a species sometimes develop different proportions than smaller ones, and that without additional material it is difficult to rule out that possibility entirely. Others have noted that the chelicera fragment, while clearly belonging to a large animal, cannot on its own confirm every detail of the size estimate. These are fair points, and the scientific community has not ignored them. But the weight of the evidence, and the consensus among those who have studied eurypterid anatomy most carefully, supports the conclusion the Jaekelopterus rhenaniar was genuinely enormous. The uncertainty is real, but it does not undermine the basic finding. It simply reminds us to hold our conclusions with appropriate care, which is in the end what science asks of us. The rock formation in which the chelicera was found is part of a broader set of Devonian-era deposits in the Rhineland-Palatinate region of Germany. The Devonian period, which lasted from roughly 419 million to 359 million years ago, left behind rock layers across parts of Europe that have been studied by geologists and paleontologists for well over a century. The region around Prüm sits within what geologists call the Rhenish Massif, an ancient uplifted block of rock that preserves sedimentary layers from a time when that part of the world looked nothing like it does today. No Rhine River, no forests, no rolling German countryside, just warm, shallow water, soft sediment, and the slow accumulation of organic material that would eventually, over hundreds of millions of years, become the stone in which Poschmann and his team found their remarkable fragment. It is worth pausing for a moment on the fact that the fossil survived at all. Fossilization is not a guaranteed process. The vast majority of organisms that have ever lived on Earth left no trace whatsoever. Soft tissues decay. Hard parts dissolve, are eaten, are scattered, are crushed by geological pressure, or are simply never buried in the right conditions to be preserved. For a fossil to form, an organism or part of an organism must be buried relatively quickly after death, in sediment that is fine-grained enough to preserve detail, in conditions where chemical processes favour mineralization rather than decomposition. For a claw fragment from an animal that lived 400 million years ago to survive intact enough to be measured and studied in a modern laboratory, a remarkable sequence of events had to occur without interruption, across an almost incomprehensible span of time. That it did is, in its quiet way, as extraordinary as the creature itself. The announcement in biological letters attracted attention well beyond the usual paleontological journals. Science journalists picked it up. Natural history museums noted it. The popular press ran headlines that were, predictably, somewhat more dramatic than the paper itself, though in this case the underlying finding was dramatic enough that the headlines were not entirely wrong. Giant sea scorpion bigger than a man, that was the phrase that travelled, and while Jaekelopterus is not technically a scorpion, as we will explore in the next part of this story, the phrase captured something real. There was, in fact, an animal of this approximate description. It was not a legend. It was not a misidentification. It was a real creature, documented by real evidence, studied by real scientists, and published in a real peer-reviewed journal. The claw in the rock was not a curiosity, it was a key. What makes the 2007 discovery particularly interesting is not just what it confirmed, but what it opened up. Before Poschmann's paper, the largest known eurypterid was generally considered to be Pterygotus anglicus, estimated at around 1 meter 75 centimeters, or just under 6 feet. That was already an impressive animal, large enough to be taken seriously as a significant predator in its environment. But Jaekelopterus, at 2.5 meters, was not simply a little larger. It was substantially larger, large enough to shift our understanding of what was biologically possible for an arthropod in a meaningful way. It pushed the upper boundary of the group outward by a significant margin and raised a whole set of new questions. Not just how large was this animal, but how did it live? What did it eat? How did it move? How did it grow to that size when every arthropod alive today is a fraction of its scale? What does its existence tell us about the world it came from? Those questions do not have simple answers, and we are going to spend a good deal of time exploring them. But they all begin with that single fragment, that 46-centimetre piece of chitin sitting quietly in a layer of Devonian stone in the Rhineland for 400 million years, waiting for someone to find it and ask the right questions. Poschmann and his colleagues found it. They asked the questions carefully, and the answers they arrived at gave us something we had not had before: a name, a measurement, and a rough outline of a creature so far outside our ordinary experience of living things that even now, knowing it was real, knowing the evidence is solid, it takes a moment to let it settle. 2.5 meters, 46-centimetre claws, an animal built from segments and shell, moving through warm, shallow water in a world that no longer exists, in a time so remote that the continents themselves were in different positions. Real, documented, studied, and in the way that all genuinely remarkable things are, still not entirely understood, because a single claw, however carefully measured and however skillfully interpreted, can only tell us so much. To understand Jaekelopterus fully, or as fully as the fossil record allows, we need to understand the broader group it belonged to: the Eurypterids, a lineage of animals that lasted for over 200 million years, produced more than 250 known species, and occupied a range of environments and ecological roles that most people have never had cause to think about. They are not famous the way dinosaurs are famous. They do not appear in children's books with the same regularity, but they are, in their own right, one of the most successful and varied groups of animals that have ever lived on this planet. And Jaekelopterus, enormous as it was only the most extreme expression of a body plan that had been quietly evolving and diversifying long before it arrived, and would continue long after it was gone. So let us talk about Eurypterids properly, because they deserve more than a passing mention. They deserve a moment of genuine attention, the kind you might give to a family you have heard of distantly but never really sat down to understand. Because once you do, once you start to see what they actually were and how long they lasted and how varied they became, the existence of something like Jaekelopterus starts to make a different kind of sense. It stops feeling like an anomaly and starts feeling like the logical, if extreme, conclusion of a very long experiment. Eurypterida is the name of the order, and the word itself comes from the Greek for broad wing, a reference to the wide paddle-like appendages that many species use for swimming. They are chelicerate arthropods, which places them in the same broad grouping as spiders, scorpions, horseshoe crabs, and mites. That word chelicerate refers to the presence of chelicerae, which are the grasping appendages near the mouth. The structures that in Jaekelopterus grew to 46 centimetres and gave paleontologists their first real measurement of the animal. Chelicerates are distinct from the other major arthropod groups, the crustaceans and the insects and the myriapods, in ways that go deeper than surface appearance. They represent a different branch of the arthropod family tree, one that diverged very early and went its own direction across hundreds of millions of years. What Eurypterids are not is worth clarifying, because the name sea scorpion, convenient as it is, carries implications that are not quite accurate. They are not true scorpions. True scorpions are arachnids, terrestrial animals with a specific body plan that includes a narrow waist, 8 legs, and the characteristic curved tail with a venomous sting. Eurypterids share a common ancestor with scorpions somewhere deep in the Cambrian or Ordovician past, but the two groups diverged long before either became what we recognise today. Eurypterids are also not crabs, not insects, not lobsters. They are their own thing, occupying a position in the arthropod family tree that is closest, among living animals, to the horseshoe crabs. If you have ever seen a horseshoe crab on a beach, with its broad domed shell and its long pointed tail and its slightly prehistoric quality that makes it look like something that wandered in from another era, then you have seen the living animal that most closely resembles the general body plan of the Eurypterids. Not identical, but related in a way that is more than superficial. The Eurypterid lineage first appears in the fossil record during the Ordovician period, roughly 460 million years ago. They reached their greatest diversity during the Silurian, persisted through the Devonian, and declined gradually through the Carboniferous and Permian, before finally disappearing at the Permian-Triassic boundary approximately 252 million years ago. That is a run of over 200 million years, too. To put that in perspective, the entire span of dinosaur existence from their first appearance to the end Cretaceous extinction covers roughly 165 million years. Eurypterids outlasted that by a significant margin. They were not a brief experiment. They were a long, sustained and genuinely successful group of animals that occupied a wide range of environments across an enormous stretch of geological time. More than 250 species are currently known, and that number continues to grow as new specimens are described. They ranged in size from small, delicate forms just a few centimetres long to the giants we're concerned with here. Most species were modest in scale, typically under 20 centimetres, which is roughly the length of a large hand. Some reached around a metre, which is already large for an arthropod by modern standards. And then there was the extreme end of the size range, where Jaekelopterus sat, alone and unreachable, at 2.5 metres. The distribution of sizes across the group suggests that gigantism was not the default condition. It was something that certain lineages achieved under certain conditions, and we will come to those conditions in time. The general body plan of a Eurypterid, once you understand it, has a kind of elegant logic to it. The body is divided into two main sections, as is common in chelicerates. The front section is called the prosoma, which functions roughly like a head and thorax combined. It is typically broad and flat, covered by a rigid shield-like plate, and it houses the animal's eyes, brain, and the bases of all its appendages. The rear section is called the opisthosoma, which is the abdomen, a long segmented structure that tapers toward the tail. At the very end of the opisthosoma is the telson, a terminal structure that in different Eurypterid species took different forms, sometimes a flat plate, sometimes a pointed spike, sometimes a broad paddle. In Jaekelopterus, the telson appears to have been a pointed structure, though the evidence for this comes from related species rather than direct fossil material. The appendages of a Eurypterid number 6 pairs in total, all attached to the prosoma. The first pair is the chelicerae, those grasping claws at the front of the head. Behind them come the other 5 pairs, which serve different functions depending on their position and the specific species involved. In most Eurypterids, the front walking legs were slender and likely used for walking along the substrate, feeling the environment and manipulating food. The rear appendages were typically the most modified, often developing into broad flattened paddles that the animal used for swimming. These paddle limbs are the broad wings that give the group its name, and in large species like Jaekelopterus, they would have been substantial structures, capable of generating real propulsive force through the water. Respiration in Eurypterids was accomplished through structures called book gills, which were located on the underside of the opisthosoma. Book gills are thin, layered structures with a large surface area for gas exchange, similar in principle to the book lungs found in modern arachnids, though adapted for aquatic rather than aerial oxygen extraction. They are called book gills because the layered structure resembles the pages of a book when viewed in cross-section, which is one of those anatomical naming decisions that turns out to be surprisingly descriptive. Horseshoe crabs still use book gills today, which gives us a living reference point for how this respiratory system works, though scaling it up to the size of Jaekelopterus raises questions we will return to later in this story. The eyes of Eurypterids sat on top of the prosoma, typically as a pair of compound eyes positioned toward the outer edges of the head shield. Compound eyes, as you may know from modern insects and crustaceans, are not like vertebrate eyes. They do not produce a single focused image in the same way. Instead, they provide a wide field of view made up of many individual light-sensing units, each capturing a slightly different angle. This gives excellent sensitivity to movement across a broad visual field, which is useful for detecting prey or predators in the water. In many Eurypterids, the eyes were positioned on the upper surface of the prosoma, suggesting that the animal spent time on or near the bottom and looked upward, which has implications for how we think about its hunting behaviour. We will come to that in detail. The exoskeleton of Eurypterids, like that of all arthropods, was made primarily of chitin, a tough lightweight biological polymer that forms the structural basis of arthropod shells everywhere from beetles to crabs to centipedes. In aquatic arthropods, the exoskeleton is often reinforced with minerals, particularly calcium carbonate, which adds rigidity and durability. The degree of mineralization varied across Eurypterid species, and this variation affected how well different species preserved as fossils. Heavily mineralized shells fossilize more readily than lightly mineralized ones, which means that our fossil record of Eurypterids may be skewed towards species with more robust exoskeletons, a point worth keeping in mind when we try to assess how diverse and widespread the group actually was. The genus name Jaekelopterus honours Otto Jaekel, a German paleontologist who lived from 1863 to 1929 and made significant contributions to the study of early vertebrates and invertebrates. Jaekel worked extensively on fossil fish, echinoderms, and other Paleozoic groups, and his name has been attached to several genera across different animal groups. The species name Rhenaniae refers to the Rhineland, the region of Germany where the fossil was found, a standard practice in paleontological naming that ties the animal to its place of discovery. So the full name, Jaekelopterus rhenaniae, carries within it both a tribute to a scientist and a geographical marker, a small piece of history embedded in the taxonomy. Within the Eurypterids, Jaekelopterus belongs to the family Pterygotidae, a group of large swimming Eurypterids characterized by their particularly well-developed chelicerae and their streamlined body forms. Other members of this family include Pterygotus and Acutiramus, both of which were also large animals, though neither reached the scale of Jaekelopterus. The pterygotids are generally considered to have been active predatory Eurypterids, more adapted for swimming in open water than for crawling along the bottom, though the degree to which any given species was truly pelagic versus bottom associated is a matter of ongoing interpretation. What they share is the general commitment to large chelicerae as the primary tool for capturing and handling prey, which tells us something about how this lineage made its living. It is worth noting that not all Eurypterids were marine or even fully aquatic. Some species appear to have lived in brackish estuaries, in freshwater lakes and rivers, or in environments that shifted between salt and fresh water depending on the season or the geography. There is even evidence that some Eurypterids may have been capable of brief excursions onto land, or at least into the intertidal zone, though they were not terrestrial animals in the way that later arachnids became. The range of environments they occupied is one of the reasons the group lasted so long. They were not locked into a single ecological niche. They were flexible, diverse, and capable of adapting to changing conditions across a wide range of habitats. What makes the Eurypterids particularly interesting from an evolutionary standpoint is their relationship to the arachnids. The prevailing view among researchers is that arachnids, the group that includes spiders, scorpions, mites, and their relatives, evolved from Eurypterid-like ancestors during the Silurian or early Devonian period. The transition from aquatic chelicerate to terrestrial arachnid was one of the great evolutionary crossings in the history of animal life, comparable in significance to the transition of vertebrates from water to land. Book gills became book lungs, paddle limbs became walking legs, aquatic hunters became terrestrial predators. The body plan that Jaekelopterus represented in its most extreme aquatic form was, in a different lineage, the starting point for the animals that would eventually become the spiders and scorpions we know today. There is something quietly remarkable about that connection. When you see a spider crossing a wall, or a scorpion held up in a field guide, you're looking at a distant relative of Jaekelopterus. Not a direct descendant, not a simplified version, but a cousin, separated by 400 million years and a fundamental shift in habitat and lifestyle. The chelicerae are still there, modified but recognisable. The segmented body is still there, reorganised but present. The basic logic of the chelicerate body plan persists across all that time and all that change, which tells us something about how durable certain evolutionary solutions can be once they take hold. The Eurypterids themselves, of course, never made that transition to land. They remained aquatic, or at least semi-aquatic, throughout their existence. And for most of their 200 million year run, that was not a limitation. The seas and estuaries and rivers they inhabited were rich, productive environments full of potential prey and ecological opportunity. It was only later, as jawed fish diversified and grew larger and more capable, and as the environments themselves began to change, that the Eurypterids found themselves under pressure they could not adapt away from. But that is a later part of the story, and we are not there yet. For now, what matters is the picture that emerges from all of this. Eurypterids were not a footnote in the history of animal life. They were a major lineage, diverse and durable, occupying a significant portion of the Paleozoic world's aquatic ecosystems. They came in many sizes and shapes and ecological roles. They were the closest ancient relatives of the animals that would become spiders and scorpions and horseshoe crabs. And at one end of their size range, at the absolute extreme of what their body plan could apparently achieve, there was Jaekelopterus rhenaniae, 2.5 meters of chelicerate arthropod, moving through the warm, shallow water of a world that no longer exists. Understanding the group is what makes the giant meaningful. Without that context, it is just a big animal. With it, it becomes something more. The furthest point reached by a lineage that had been quietly building towards something extraordinary for tens of millions of years. And to understand just how extraordinary that body was, we need to look at it directly. Not as a set of measurements and ratios, but as a physical object in the world. A creature with a shape and a weight and a texture and a presence. Because the numbers are one thing. But picturing what Jaekelopterus actually looked like, what it felt like to be in the water near it, what it would have meant to be something smaller moving through the same environment, that is where the story becomes truly vivid. So let us look at it directly. Let us walk through the body of Jaekelopterus rhenaniae from front to back, the way you might study any large animal, carefully and slowly, paying attention to the details that make it real rather than simply impressive. We begin at the front where the chelicerae are. These are the great grasping claws that define this animal in the fossil record, and that gave us our first and most reliable measurement. Each chelicera was approximately 46 centimeters long. That is not a small number to hold in the mind, so it helps to reach for a comparison. Hold your arm out in front of you. The distance from your shoulder to the tips of your outstretched fingers, on most adult humans, is somewhere between 60 and 70 centimeters. The chelicera of Jaekelopterus, a single claw, came close to matching that length. Not the whole arm, just the claw. And there were two of them, one on each side of the front of the animal, working together to grasp and manipulate whatever Jaekelopterus had decided to eat. They were not delicate structures. They were built for strength. The segments of the chelicera were heavily reinforced, and the tips likely came to sharp points or edges capable of gripping and piercing prey. In the pterygotid Eurypterids generally, the chelicerae are thought to have been the primary tools of predation, used to seize prey and hold it while the animal fed. In Jaekelopterus, those tools were simply larger than in anything else we have found. Behind the chelicerae sits the prosoma, which is the front body section, roughly equivalent to what we might call the head and thorax combined. In Jaekelopterus, The prosoma would have been a broad, relatively flat shield-like structure, wider than it was tall. This shape is consistent with other pterygoid eurypterids and reflects a body plan that was built for a life spent moving through water rather than standing on land. The prosoma housed the brain, the primary sensory organs, and the attachment points for all 6 pairs of appendages. On the upper surface, positioned toward the front and slightly to the sides, were the compound eyes. These were not small or subtle. Compound eyes in eurypterids were often large relative to the prosoma, and in an animal the size of Jaekelopterus, they would have been substantial structures. Their position on the top of the prosoma is significant. Animals with eyes positioned on the upper surface of a flat head tend to be oriented toward what is above them, which fits well with a predator that may have lurked near the bottom of shallow water, watching the world above for the silhouettes of potential prey moving through the water column. There is also a possibility that Jaekelopterus had a pair of smaller median eyes near the centre of the prosoma, in addition to the large lateral compound eyes. This is consistent with other eurypterids, and the median eyes may have functioned as simple light detectors rather than image-forming structures, helping the animal orient itself relative to light and shadow. From the prosoma extended 6 pairs of appendages, and these are worth going through with some care because they were not all the same. The front pairs were walking and sensory limbs, relatively slender, jointed in the way you might expect from an arthropod, and used for moving along the substrate and perhaps for manipulating small objects or food. As you move toward the back of the prosoma, the appendages change character. The rearmost pair, the 6th pair, were the swimming appendages, and these were dramatically different from the walking legs. They were broad, flattened and paddle-like, sometimes described as looking like the blade of an oar. These paddle limbs were the primary propulsive structures used to drive the animal through the water with powerful strokes. In a creature the size of Jaekelopterus, those paddles would have been large enough to generate considerable thrust, though as we will consider later, the animal was probably not a fast sustained swimmer so much as a capable one when it needed to be. The intermediate appendages, the ones between the front walking legs and the rear paddles, were likely a mix of sensory and locomotive function, helping the animal navigate, feel its environment, and perhaps assist in handling prey once the chelicerae had made their initial capture. Moving behind the prosoma, we reach the opisthosoma, which is the abdomen, the long segmented rear portion of the body. In Jaekelopterus, as in other eurypterids, the opisthosoma was divided into segments that gradually narrowed toward the back. The front segments of the opisthosoma were broader and housed the book gills, which were the respiratory organs. Book gills are layered, leaf-like structures that extract oxygen from the water, and they are the same structures that, in a different evolutionary line, eventually became the book lungs of terrestrial arachnids. In Jaekelopterus, the book gills would have needed to be substantial to supply oxygen to a body of this size, and there is genuine scientific interest in whether they could have done so efficiently enough to support very active behaviour. This is a question we will return to because it bears directly on what kind of life Jaekelopterus was actually capable of living. The posterior segments of the opisthosoma became progressively narrower and more tapered, giving the animal a somewhat elongated, slightly wedge-shaped silhouette when viewed from above. At the very end of the opisthosoma was the telson, a tail-like spike that projected from the rear of the body. In Jaekelopterus, the telson was a rigid, pointed structure. It was not a stinger in the way a scorpion's tail is a stinger. There is no evidence of a venom gland associated with the telson of eurypterids. It may have served a stabilizing function during swimming, helping the animal maintain its orientation in the water, or it may have had some role in anchoring the animal to the substrate. It is also possible that it was simply a structural remnant of the ancestral body plan, retained without a strong specific function. These are the kinds of questions that a single claw fragment cannot answer. Now, what did all of this add up to in terms of physical scale? The estimated total body length of Jaekelopterus, derived from the proportional scaling of the chelicera, is approximately 2.5 meters. That is roughly 8 feet and 2 or 3 inches, depending on which conversion you prefer. To picture that in practical terms, The average doorway in a modern home is about 2 meters tall. Jaekelopterus would not have fit through it. It would have needed 0.5 meter more of clearance just to pass through without its telson scraping the frame, and that assumes it was moving in a straight line, which a living animal rarely does. Laid out on a sidewalk, it would have occupied the space of a large adult human plus a small child standing behind them. That is a substantial animal by any measure. The question of weight is harder to answer than the question of length, because weight depends on the density and thickness of the body, not just its linear dimensions. Arthropods do not scale the same way vertebrates do, and the exoskeleton of a large eurypterid would have had a different mass distribution than the skeleton of a large fish or mammal. Some researchers have attempted rough estimates based on comparisons with other large arthropods and with the known proportions of smaller eurypterids. The figures that emerge from these calculations are in the range of 150 to perhaps 200 kilograms, though these are genuinely rough estimates and should be treated as approximations rather than precise measurements. What they suggest is that this was not a lightweight animal. It was not the kind of creature that moved quickly and effortlessly through the water without effort. It was a heavy, substantial organism that would have displaced a meaningful volume of water as it moved, and that would have required real muscular power to propel itself. The exoskeleton itself deserves a moment of attention. Like all arthropods, Jaekelopterus was encased in an exoskeleton made primarily of chitin, the same tough biological polymer that forms the shells of crabs and lobsters, and the wings of beetles. In aquatic arthropods, the chitin of the exoskeleton is often reinforced with mineral deposits, particularly calcium phosphate or calcium carbonate, which add rigidity and hardness. The exoskeleton of Jaekelopterus would have been a substantial piece of biological engineering, thick enough to provide protection from other predators and from the mechanical stresses of moving through water, but not so thick as to make the animal immobile. There is an inherent tension in the design of any large arthropod exoskeleton. Too thin and it fails to protect, too thick and the animal cannot move. The eurypterids apparently found a workable balance, at least for the environments they occupied. In water, the buoyancy of the surrounding medium helps support the weight of the exoskeleton, which is one reason why very large arthropods have historically been aquatic. On land, the same exoskeleton would have been far more burdensome, and the muscles required to move it would have had to work against gravity rather than with the assistance of buoyancy. Jaekelopterus was a creature of the water in a very fundamental sense. As for coloration, we have no direct evidence. Soft tissues, including pigmentation, do not preserve in the fossil record under normal circumstances. What we can do is look at the coloration of related modern animals and at the general principles of how aquatic predators tend to be coloured and make an educated guess. Modern horseshoe crabs, which are among the closest living relatives of eurypterids, are a dark olive-brown colour that blends reasonably well with sandy and muddy substrates. Many aquatic ambush predators tend toward mottled or counter-shaded patterns, darker on top and lighter underneath, which helps them blend into the bottom when viewed from above and into the lighter water surface when viewed from below. It is entirely plausible that Jaekelopterus was similarly coloured, perhaps a dark brownish-green or grey-brown, with some degree of mottling or patterning that helped it blend into the shallow, sediment-rich environments it likely inhabited. But this is speculation, offered in the spirit of trying to make the animal feel real, not as a claim about what we actually know. The honest answer is that we do not know what colour it was, and we may never know. What we can say with more confidence is that Jaekelopterus was a visually striking animal in purely physical terms. 2.5 meters of segmented body, broad and flat at the front, tapering toward the rear, with a pair of massive claws extending forward and a pair of broad paddle limbs sweeping at the back, moving through shallow water with the slow, deliberate authority of something that did not have many reasons to hurry. The compound eyes on the top of the prosoma would have scanned the water above as the animal rested on or near the bottom. The chelicerae would have been held slightly forward, ready. The paddle limbs would have made small adjustments to maintain position in the current. It would not have looked frantic or rushed. Large predators rarely do. They tend to look patient, and patience in a large predator is a kind of threat all by itself. There is one more comparison worth making before we move on, because it helps anchor the scale of this animal in a way that numbers alone sometimes cannot. The chelicera of Jaekelopterus, at 46 centimetres, is roughly the length of a standard school ruler, plus about half another one. That is the measurement of a single claw. The animal that claw belonged to was more than 5 times that length from front to back, and that animal was moving through real water, in a real ecosystem, interacting with real prey and real competitors, in a world that was as fully alive and complex as the world we inhabit today, even if it was populated by entirely different kinds of life. The fragment found in Germany in 2007 is not just a measurement, it is a window into a body, and that body was part of a world we're only beginning to understand in any real detail. And to understand that world, we need to step back from the anatomy and look outward at the environment that shaped Jaekelopterus, and that Jaekelopterus in turn helped to shape. Because the creature does not make full sense without the water it moved through, the sea floor it hunted over, and the other animals it shared that ancient world with. The world that Jaekelopterus moved through was not the world we know. It was not even close. To stand at the edge of that ancient sea, if such a thing were somehow possible, would be to feel the deep strangeness of deep time pressing against you from every direction. The sky above would have been the right colour, roughly, blue and bright, with clouds moving across it in patterns that would feel almost familiar. But everything below the horizon would have been wrong in ways that would take a moment to fully register. Not wrong in the sense of being broken or lesser. Wrong in the sense of being genuinely alien, a world still in the process of becoming the world we eventually inherited, 400 million years away from the moment you are reading or listening to this. The time period we're talking about spans what geologists call the late Silurian and early Devonian, a stretch of Earth history running from roughly 432 million years ago to approximately 393 million years ago. Those numbers are large enough to be difficult to hold in the mind all at once, so it helps to approach them from a different angle. Modern humans, as a species, have existed for perhaps 300,000 years. The gap between us and Jaekelopterus is more than 1,000 times that. Whatever we think of as old, whatever feels ancient to us, whether it is a medieval castle, a Roman road, an Egyptian pyramid, or even the earliest cave paintings, all of it is compressed into a tiny sliver at the very end of the timeline that separates us from this animal. When Jaekelopterus was alive, the rocks that would eventually become those pyramids had not yet formed. The continents had not yet arranged themselves into anything we would recognise. The geography of the late Silurian and early Devonian world was dramatically different from our own. The two major land masses of the time were Gondwana, a vast southern supercontinent that contained much of what would eventually become South America, Africa, Antarctica, Australia, and the Indian subcontinent, and Laurussia, sometimes called Euramerica, which contained the ancestral cores of North America, Europe, and parts of Asia. These land masses were positioned very differently from where their descendants sit today. What is now Central Europe, including the region of Germany, where the Jaekelopterus fossil would eventually be found, was located close to the equator during this period. Not near the equator in the sense of being warm and tropical in the way we might picture a modern tropical coast, but genuinely equatorial, sitting at low latitudes where solar energy was intense and the climate was correspondingly warm. The shallow seas that covered much of this region were warm in a way that modern temperate European coastlines are not, more like the warm, shallow coastal waters of a modern tropical or subtropical zone than anything you would encounter off the coast of Germany today. These were not open ocean environments, at least not primarily. The environments where Eurypterids like Jaekelopterus are most commonly found in the fossil record tend to be brackish or near-shore marine settings, coastal lagoons, estuaries, shallow embayments, and the kinds of environments where freshwater and saltwater mix in varying proportions. Some researchers have debated whether Jaekelopterus and its relatives were fully marine, primarily freshwater, or tolerant of a wide range of salinities. The evidence from the rock formations where Eurypterid fossils are found suggests that many species, including some of the largest, preferred brackish or nearshore conditions rather than the open deep ocean. This makes a certain ecological sense. Shallow coastal environments tend to be richer in food resources than open water. Sediment accumulates on the bottom, providing habitat for smaller invertebrates. Algal mats grow in the shallows, supporting a base of primary productivity. Prey animals concentrate in these areas, and a large predator would logically follow its prey. The sea floor in these environments was not bare sand or bare rock. It was soft, often muddy sediment, rich in organic material, covered in places by thick mats of algae and early microbial communities. Early coral-like reef structures were beginning to form during this period, though the reefs of the Silurian and Devonian were built not by the stony corals we associate with modern reefs, but by tabulate and rugose corals, along with stromatoporoids, which were sponge-like organisms that built large mounded calcareous structures. These reef communities provided complex three-dimensional habitat, full of crevices and overhangs where smaller animals could shelter, and where a large predator might find hunting opportunities. The water column above the sediment would have been reasonably clear in some areas, and turbid with suspended particles in others, depending on currents, river input, and biological activity. The atmosphere of the late Silurian and early Devonian was different from today's, in ways that would have mattered to any animal living in or near it. Oxygen levels during this period were somewhat lower than the 21% we breathe today, possibly in the range of 15 to 17% by some estimates, though the precise figures are debated and vary depending on the model used to reconstruct ancient atmospheric composition. This is worth noting because it complicates some of the explanations for why arthropods grew so large during the Paleozoic, which we will return to later in this story. Carbon dioxide levels were significantly higher than today, which contributed to a generally warmer global climate. There was no permanent polar ice to speak of during much of this period, and sea levels were correspondingly higher than today. The world was, in broad terms, warmer, wetter in some regions, and more uniformly distributed in its climate, without the sharp temperature gradient between equator and poles that characterises the modern Earth. On land, the picture was sparse by any standard we're used to. There were no trees in the early Devonian in the sense we understand trees today. The land surface near the shores of these ancient seas was colonised by early plant life, but it was modest and low-growing. Mosses and liverworts clung to damp surfaces near the water's edge. Early lycopsides, the ancestors of club mosses, were beginning to develop upright growth forms, but they were small. The first truly vascular plants, those with internal systems for transporting water and nutrients, were just beginning to diversify during this period. The Devonian is sometimes called the Age of Fishes, but it could equally be called the Age of the First Forests, because it was during the Devonian that land plants began their slow transformative expansion across the terrestrial surface. At the start of the period, however, the land would have looked more like a coastal mudflat colonised by low green crusts and small leafless stems than anything resembling a forest. There were no grasses, no flowering plants, no ferns in the familiar sense. The green edge of the land was thin and low, and beyond it, moving inland, the rock was largely bare. There were no large animals on land at all during the early Devonian. The colonisation of terrestrial environments by vertebrates was still tens of millions of years in the future. There were no insects with wings, no reptiles, no amphibians in any recognisable form. Small primitive arthropods, some of the earliest land-dwelling invertebrates, were beginning to venture onto moist terrestrial surfaces, but they were tiny and ecologically marginal. The sound of the land, if you could somehow hear it, would have been wind, rain, the movement of water, and nothing else. No bird song, no insect chorus, no rustling of large animals through undergrowth. The terrestrial world was quiet in a way it has not been since. All of the ecological action, all of the biological complexity and diversity was in the water. And the water was full of life, much of it unfamiliar by modern standards, but rich and varied and organised into genuine food webs, with clear trophic levels. The base of the aquatic food web rested on algae, cyanobacteria, and other photosynthetic organisms that were abundant in the shallow, well-lit coastal waters. Above them were the filter feeders and detritivores, animals that fed on suspended particles or on the organic material accumulating in the sediment. Trilobites were still present during this period, though they were past their peak diversity. Many trilobite species were bottom dwellers that moved through the sediment in search of food, and they would have been a familiar presence on the seafloor in the environments where Jaekelopterus lived. Brachiopods, which are shelled animals superficially resembling clams but belonging to a completely different phylum, were extremely abundant and diverse. Early cephalopods, ancestors of modern nautiluses and squids, moved through the water column, using jet propulsion to hunt smaller prey. The vertebrates of this world were very different from the fish we know today. The late Silurian and early Devonian seas were populated primarily by jawless fish, the agnathons, which included animals like ostracoderms, heavily armoured, bottom-dwelling creatures that fed on organic material in the sediment, and more active forms that swam in the water column. These early fish had no movable jaws in the modern sense. They had mouths adapted for filter feeding or suction feeding, and while some of them were armoured with bony plates, they were not large animals by the standards of what Jaekelopterus could have encountered. Jawed fish, the group that would eventually give rise to sharks, bony fish, and ultimately all land vertebrates, were diversifying rapidly during the Devonian. Early sharks, small and different in many respects from their modern descendants, were present. Placoderms, a now extinct group of armoured jawed fish that would eventually include some of the largest predatory fish of the Devonian, were beginning their radiation. But in the earlier part of the period, when Jaekelopterus was at its peak, many of these vertebrate lineages were still relatively small and ecologically limited compared to what they would later become. This is a point worth sitting with for a moment, because it matters for understanding the ecological position of Jaekelopterus. The vertebrate revolution, the eventual dominance of fish and their descendants over aquatic and terrestrial ecosystems, had not yet fully arrived. The large vertebrate predators that would later come to dominate marine food webs, the big placoderms like Jaekelopterus with its metre-wide armoured head, the large sharks, the predatory lobe-finned fish that would eventually give rise to land animals, these were either absent or still in early stages of their evolution during the time when Jaekelopterus was alive. Into that ecological space, the Eurypterids had expanded. They had diversified into many different niches, and the largest of them, Jaekelopterus at the extreme end, occupied a position near or at the top of the food web in the environments it inhabited. Not because it had outcompeted vertebrate predators in some dramatic confrontation, but because those vertebrate predators had not yet grown large enough or diverse enough to fill that role themselves. It is also worth noting that Jaekelopterus shared its world with other Eurypterids, including some that were themselves quite large. The genus Pterygotus, which is closely related to Jaekelopterus and belongs to the same family, Pterygotidae, reached estimated lengths of around 1 3/4 meters. Acutiramus, another pterygotid, reached approximately 2 meters. These animals were not small, and they were not ecologically trivial. Whether Jaekelopterus competed directly with them, preyed upon them, or occupied sufficiently different microhabitats to avoid direct competition is not something we can determine from the fossil record with any confidence. What we can say is that the Eurypterid fauna of the late Silurian and early Devonian included multiple large species, and that the ecosystems of this period were capable of supporting that level of large predatory arthropod diversity. The water that these animals moved through was warm, shallow in many places, and rich in the kinds of organic productivity that support complex food webs. The seafloor was soft and navigable. The water column above it was populated by a diverse array of invertebrates and early vertebrates. The edges of the sea were fringed by early plant life and the first tentative experiments in terrestrial colonization. It was a world in transition, moving from the ancient marine-dominated ecosystems of the Ordovician and Silurian toward the more familiar vertebrate-dominated world of the later Devonian and beyond. Jaekelopterus lived in the middle of that transition, large and well adapted to its environment, a product of hundreds of millions of years of arthropod evolution, and also, though it could not have known this, living in the last chapter of the era when arthropods truly ruled the largest ecological roles in complex aquatic ecosystems. Understanding the environment is not just background detail, it is the context that makes the animal's body make sense. Those broad paddle limbs were shaped by the need to move through shallow, sometimes turbid water. Those compound eyes positioned on the top of the head were shaped by the need to scan the water above while resting near the bottom. Those massive chelicerae were shaped by the prey available in these rich, shallow, warm coastal seas. The environment and the animal are not separate things. They are a conversation, written in anatomy and geology and time. And knowing something about the world Jaekelopterus inhabited brings us naturally to the next question, the one that perhaps feels the most immediate and the most vivid. What exactly did it do in that world, and how did it hunt? That question, what Jaekelopterus actually did in its world, how it moved and fed and positioned itself within the living web of its environment, is one that paleontologists can only answer indirectly. There is no behavioural record. There are no footprints that tell us how it approached prey, no stomach contents preserved in the rock, no trace fossils that clearly document a hunt in progress. What we have instead is anatomy. An anatomy, read carefully and compared thoughtfully to living animals, can tell us quite a lot. It is not the same as watching the animal move, but it is not nothing either. It is a kind of inference that paleontologists have become skilled at making, and the picture it produces, while always provisional, is detailed enough to be genuinely illuminating. The first and most important piece of evidence is the chelicerae themselves. Those enormous grasping claws, each estimated at around 46 centimetres in length, are not the anatomy of a passive animal. They are not the anatomy of a filter feeder, which would have no need for powerful grasping structures at the front of the body. They are not the anatomy of a detritivore, an animal that feeds on organic material in the sediment, which would require a very different kind of mouthpart arrangement. Large, robust, articulated chelicerae of the kind found in Jaekelopterus are the anatomy of an active predator. They are structures built to seize, to hold, to restrain. In living chelicerates, the closest relatives of eurypterids, chelicerae serve exactly this function. Spiders use them to inject venom and manipulate prey. Horseshoe crabs use smaller versions to handle food items near the mouth. The scorpions, which are the living animals most visually reminiscent of eurypterids, use their pedipalps, a different pair of appendages, to grasp prey. But the functional principle is similar. A large, well-developed grasping structure at the front of the body is a predatory tool. In Jaekelopterus, those tools were enormous. The positioning of the eyes provides another important clue. The compound eyes of Jaekelopterus were located on the top of the broad, flat prosoma, the head shield, rather than on the sides or front. This is a placement that appears repeatedly in animals that hunt from below, or that rest on or near the substrate and watch for movement above them. Crocodilians have their eyes positioned on the top of the head, allowing them to remain almost entirely submerged while still scanning the water surface and the banks above. Flatfish have eyes that migrate to the top of the head as they mature, allowing them to lie on the sea floor and watch for prey above them. The logic is the same in each case. If your body is low and your prey is above you, you want your eyes pointing upward. For Jaekelopterus, resting on or near the sea floor in shallow coastal water, eyes on top of the prosoma would have allowed it to monitor the water column above while remaining relatively still and inconspicuous against the substrate. This is the anatomy of an ambush predator, or at the very least, an animal that relied heavily on stillness and position rather than active pursuit. This brings us to the question of locomotion, because how Jaekelopterus moved tells us a great deal about what kind of hunter it could have been. The 6 pairs of appendages in Eurypterids serve different functions. The front pairs were used for walking and sensory exploration of the substrate. The rear appendages, in pterygotid Eurypterids like Jaekelopterus, were modified into large, flattened paddle-like structures, sometimes described as swimming paddles or natatory appendages. These paddles were clearly adapted for swimming, and in related species they are well preserved enough to show their broad, leaf-like shape. The presence of these paddles tells us that Jaekelopterus was capable of swimming and capable of generating significant thrust through the water, but the shape and placement of these paddles, combined with the overall body plan, suggests a specific kind of swimming rather than a general one. Large broad paddles are good for generating powerful bursts of acceleration. They are not the anatomy of an animal built for sustained, efficient long-distance swimming. A tuna, built for endurance and speed over distance, has a very different tail and body shape, streamlined, stiff, with a crescent-shaped tail that minimizes drag and maximizes propulsive efficiency. Jaekelopterus, with its broad, segmented body and large paddle appendages, was more likely built for short, powerful lunges rather than extended pursuit. Think less of a dolphin chasing prey across open water and more of a crocodile launching itself from a resting position, or a large cuttlefish using its fins to hover and then striking with sudden speed. The burst capability was probably impressive, the sustained speed was probably not. This combination of features, ambush positioning, eyes on top of the head, powerful grasping chelicerae, and a body built for short bursts rather than sustained pursuit, points toward a hunting strategy that relied on patience and proximity rather than active chasing. The most likely scenario, based on what the anatomy suggests, is that Jaekelopterus spent significant time resting on or near the sea floor, partially concealed by sediment or low-lying substrate, watching the water above it. When a suitable prey item came within range, it would have launched itself upward or forward in a short, powerful strike, seizing the prey with its chelicerae before it could escape. This is a hunting strategy that has evolved independently in many different lineages across the history of life, because it is energetically efficient and effective in environments where prey is abundant but mobile. What did it hunt? Here the inference becomes somewhat more speculative, but still grounded in what was available. The fish of the late Silurian and early Devonian, the jawless agnathons, the early jawed fish, the small early sharks, were all potential prey items for an animal of Jaekelopterus' size. A 2.5 metre Eurypterid with 46 centimetre claws would have been capable of seizing fish of considerable size, and in the environments where Jaekelopterus lived, fish of that era were generally not large. Many of the jawless fish were under 0.5 metre in length. Even the early jawed fish of this period were, in most cases, relatively modest in size compared to what they would later become. A fish of 30 or 40 centimetres, moving through shallow water above a resting Jaekelopterus, would have been a very manageable prey item for an animal with claws nearly as long as the fish itself. Smaller Eurypterids were also likely prey. Cannibalism and predation within the same broad group is not unusual in arthropods. Modern scorpions will eat smaller scorpions. Larger spiders consume smaller ones. In an environment populated by multiple Eurypterid species of varying sizes, a large Jaekelopterus encountering a smaller Eurypterid of perhaps 30 or 40 centimetres would almost certainly have treated it as a food opportunity rather than a social encounter. The chelicerae were well suited to grasping and manipulating prey with an exoskeleton, and the hard outer covering of an arthropod, while protective against some threats, would not have been an insurmountable obstacle for an animal with the mechanical advantage of those large claws, Soft-bodied prey, cephalopods, worms, early soft-bodied invertebrates would also have been accessible and energetically rewarding. The seafloor environments of the late Silurian and early Devonian were rich in soft-bodied life, much of which does not preserve well in the fossil record but was certainly present. An opportunistic predator of Jaekelopterus' size would not have been selective in a narrow sense. It would have eaten what was available, what came within range, and what could be subdued. Some researchers have raised the possibility that Jaekelopterus may also have scavenged. This is a reasonable suggestion, and it is worth taking seriously. Large predators in many ecosystems are not exclusively active hunters. They supplement their diet with carrion when it's available, because carrion is essentially a free meal that requires no expenditure of energy to subdue. A dead or dying fish, a molting eurypterid temporarily vulnerable without its hardened exoskeleton, an animal stranded by a receding tide, all of these would have been attractive food sources for an animal that needed to sustain a body of potentially 150 kilograms or more. The energetic demands of a body that large are substantial, and an animal that can exploit multiple food sources, active prey, scavenged carrion, and perhaps even plant material or invertebrates in the sediment, is better positioned to meet those demands than one that depends exclusively on successful active hunts. This brings us to a question that researchers have debated with genuine interest. Was Jaekelopterus truly an apex predator in the strict sense? Or was it better described as an opportunistic generalist that happened to be very large? The distinction matters because it changes how we understand the animal's role in its ecosystem. A true apex predator, an animal at the top of the food web with no natural predators of its own, and a hunting lifestyle that actively shapes the behaviour of prey populations, plays a specific ecological role. An opportunistic generalist of large size plays a somewhat different role, one that involves more flexibility, more variation in diet, and potentially more ecological overlap with other large animals. The honest answer is that we cannot be certain. The fossil record does not give us the behavioural resolution needed to settle this question definitively. What we can say is that the anatomy of Jaekelopterus is consistent with active predation as a significant part of its lifestyle, and that in the ecosystems it inhabited during the period it lived, there were few if any other animals that could have posed a serious threat to a fully grown adult. The large placoderms, the armored-jawed fish that would eventually include animals like Jaekelopterus, a predator with a head the size of a small car and bite force, estimates that dwarf almost anything else in the fossil record, were either not yet present or not yet large enough to compete with or predate upon Jaekelopterus during the animal's peak. In that sense, a fully adult Jaekelopterus in its prime was probably not something that other animals of the same period routinely hunted. There is one behavioural analogy that researchers have found particularly useful when thinking about Jaekelopterus, and that is the crocodilian. Modern crocodiles and alligators are large, semi-aquatic ambush predators with a body plan that shares some functional similarities with what we infer for Jaekelopterus. They rest in shallow water or at the water's edge, eyes at or above the surface, patient and still for long periods. When prey comes within range, they strike with explosive speed and hold on with powerful jaws. They are not built for long pursuit, but they do not need to be. Their hunting strategy is built around positioning, patience, and a decisive strike. They are also opportunistic, eating a wide variety of prey and not passing up a scavenging opportunity when one presents itself. The parallel is not perfect, because crocodilians are vertebrates with very different anatomy and physiology, but as a functional model for thinking about how a large bottom-associated ambush predator might have operated in a shallow water environment, it is a useful frame. The mantis shrimp offers another interesting comparison, not for size, but for the mechanics of a sudden, powerful strike from a relatively stationary position. Mantis shrimp are crustaceans, not chelicerates, and they are much smaller than Jaekelopterus, but they demonstrate how effective a strategy of concealment, followed by explosive close-range attack, can be for an arthropod predator. The mantis shrimp strike is one of the fastest movements in the animal kingdom, and it is powered by a stored energy mechanism in the appendages. Jaekelopterus would not have had the same mechanism, but the principle of a sudden, high-force strike from close range is one that the anatomy supports. What emerges from all of this is a picture of an animal that was not a relentless, high-speed pursuit predator, but was instead a patient, powerful, position-dependent hunter. An animal that used its size, its camouflage potential against the sea floor, and the upward orientation of its eyes to wait for prey to come to it, and then to seize that prey with claws large enough to be unambiguous about the outcome. An animal that probably also took advantage of whatever food sources the environment offered, including carrion and smaller, slower invertebrates. An animal that, in its ecosystem and its time, had very little to fear from other predators once it had reached full adult size, and that occupied a position at or very near the top of the food web in the warm, shallow coastal seas of the late Silurian and early Devonian. It is a compelling picture, and it is worth acknowledging even as we sit with it that it is a reconstruction. Every detail of Jaekelopterus's behaviour is inferred rather than observed. The anatomy gives us strong hints, and the comparison to living animals gives us plausible models, but the animal itself has been gone for nearly 400 million years, and the gap between inference and certainty is real. What the anatomy cannot tell us is how often it hunted successfully, how much time it spent resting versus moving, whether it was solitary or whether individuals tolerated each other's presence in the same stretch of water, whether juveniles and adults occupied the same habitats or separated by size, or any of the finer-grained behavioural details that make a living animal something more than a body plan. Those details are lost, and honestly, that loss is part of what makes the animal feel so distant and so fascinating at the same time. But even the broad picture is remarkable enough. A 2.5-metre arthropod, patient in the sediment, watching the water above with eyes built for exactly that purpose, and capable of striking with claws nearly 0.5 metre long. It is a predator that the world has not seen anything quite like since, and understanding how it hunted is part of understanding why it was possible at all. And that question, why the ancient world could produce animals of this size and this kind, why Jaekelopterus could exist at the scale it did, leads us directly into one of the most genuinely interesting debates in paleontology. The question of how we actually measure and verify the size of an animal known only from fragments, and why the ancient world seemed so willing to build arthropods so much larger than anything alive today. That question of size is worth sitting with for a moment, because it is not as simple as it might first appear. When a paleontologist holds a fossil fragment and announces a body-length estimate, the number that gets reported in a journal or picked up by a headline feels authoritative, precise, almost as if someone measured the animal directly. But the reality of how that estimate is constructed is considerably more nuanced, and understanding the methodology helps us appreciate both how confident we can reasonably be and where genuine uncertainty still lives. The estimate for Jaekelopterus rhenanii, that figure of approximately 2.5 metres, rests on a specific technique called proportional scaling, and it begins with that single Helicera fragment recovered from the Devonian era rocks near Prüm in the Rhineland-Palatinate region of Germany. The fragment measures 46 centimetres, which is already remarkable on its own. A claw nearly 0.5 metre long is not a subtle piece of anatomy, but a claw is not a whole animal, and to get from one to the other, researchers need a reliable relationship between the size of the Chelicera and the size of the body that carried it. To establish that relationship, scientists turn to related Eurypterid species for which more complete fossil material is known. The genus Pterygotus is one of the most important reference points here. Pterygotus anglicus, a well-studied Eurypterid from the Silurian period, is known from enough material that researchers have been able to construct reasonably detailed body reconstructions. For Pterygotus, the relationship between chelicera length and total body length has been measured and documented across multiple specimens. Another important reference genus is Acuteramus, a close relative of Pterygotus within the Pterygotid family, which Jaekelopterus also belongs to. Acuteramus is estimated to have reached body lengths of around 2 metres, and its chelicera to body proportions provide another data point for the scaling calculation. The logic works like this. If you know that in Pterygotus and Arthropods, the chelicera represents roughly a certain fraction of total body length, and if Jaekelopterus shares the same basic body plan and belongs to the same family, then you can apply that fraction to the 46 cm chelicera and arrive at a total body length estimate. When Marcus Poschmann and his colleagues published their findings in the journal Biological Letters in 2007, that calculation produced the 2.5 metre figure, and it was a figure that immediately placed Jaekelopterus at the top of the known Eurypterid size range, and indeed at the top of the known arthropod size range across all of Earth's history. But here is where the scientific conversation becomes genuinely interesting, and where a degree of careful skepticism is both appropriate and intellectually honest. The assumption underlying proportional scaling is that the ratio of chelicera length to body length is consistent across members of the same family, and that it scales linearly as the animals get larger. Both of those assumptions are reasonable, but neither is guaranteed. Biological scaling is not always linear. In many animal groups, certain body parts grow disproportionately as the animal gets larger, a phenomenon called allometric scaling, and there is no certainty that the chelicera of Jaekelopterus scaled in exactly the same proportion as those of its smaller relatives. If the chelicerae of the largest individuals were proportionally larger than in smaller animals, the 2.5 metre estimate might actually be an underestimate. If the chelicerae were proportionally smaller, the estimate might be too generous. Without more complete fossil material, this question cannot be resolved with full confidence. Some researchers have raised this point not to dismiss the estimate, but to flag its assumptions. The 2.5 metre figure is the best estimate available given the evidence, and it is built on a sound methodological foundation, but it carries a margin of uncertainty that the headline number does not always convey. A more conservative reading of the same data might place the animal closer to 2 metres, while a more liberal interpretation might push it toward or even slightly beyond 2.5. The published figure sits at what the available comparisons suggest is the most defensible midpoint. It is also worth noting that Jaekelopterus is not the only giant Eurypterid in the fossil record, and comparing the candidates helps put the size debate in context. Pterygotus anglicus, the species used as one of the primary scaling references, is itself estimated at around 1.75 metres, which would make it a large animal by almost any standard outside of vertebrate megafauna. Arthropods, as mentioned, reaches approximately 2 metres in some estimates. Both of these animals are known from considerably more complete material than Jaekelopterus, which gives their size estimates a somewhat firmer footing. And yet even for these better-known species, size estimates have been revised over time as new specimens have been found and methodologies have been refined. The history of Eurypterid size estimates is one of gradual recalibration, with numbers shifting as the evidence base grows. What this means is that Jaekelopterus holds the record for the largest known arthropod, but it holds that record on the basis of a single fragment, and the record itself is a calculated estimate rather than a direct measurement. That is not a reason to dismiss the finding. It is a reason to hold it with the kind of careful appreciation that good science deserves. The evidence is real, the methodology is sound, the estimate is well reasoned, and no other known arthropod fossil comes close to challenging it. But it is an estimate, and the honest acknowledgement of that fact is part of what makes the science trustworthy. There is a broader question that the size debate opens up, and it is one that paleontologists and evolutionary biologists have found genuinely fascinating for decades. Why were arthropods so much larger in the Paleozoic era than they are today? Why did the ancient world produce animals like Jaekelopterus at all? And why has nothing like them existed since? This is not a trivial question, and the answers that have been proposed are illuminating precisely because none of them is entirely satisfying on its own. The most frequently cited explanation is the oxygen hypothesis, and it has a certain intuitive appeal. The idea is that higher atmospheric oxygen levels in the ancient world would have allowed arthropods to grow larger, because arthropod respiratory systems, which rely on diffusion through book gills or tracheae, rather than an active circulatory system carrying oxygen to tissues, are limited in their efficiency at larger body sizes. More oxygen in the atmosphere or dissolved in water would extend the range over which diffusion-based respiration could support larger bodies. The Carboniferous period, which followed the Devonian, is the classic example cited in this context, because atmospheric oxygen during the Carboniferous is estimated to have reached approximately 35%, compared to today's 21%. That period produced Meganeura, the giant dragonfly with a wingspan of 65 to 70 centimetres, and Arthropods, the giant millipede estimated at up to 2.5 metres in length, which would make it the largest known land arthropod in Earth's history. But here is the complication. Jaekelopterus lived in the Silurian and early Devonian, and the oxygen levels during that period were not dramatically elevated in the same way. Estimates for atmospheric oxygen during the late Silurian and early Devonian place it at roughly 14 to 16%, actually somewhat lower than today's levels. If oxygen availability was the primary driver of arthropod gigantism, Jaekelopterus should not have been possible. The fact that it existed anyway suggests that the oxygen hypothesis, while perhaps relevant for the Carboniferous giants, does not fully explain the Eurypterid giants of the earlier Paleozoic. This is where the aquatic buoyancy argument becomes important. Water provides a fundamentally different mechanical environment than air. An animal living in water is partially or fully supported by the medium it inhabits, which means the structural demands on its skeleton are reduced compared to a land animal of the same mass. For an arthropod, this matters enormously, because the exoskeleton that provides structural support also has to be carried and moved. On land, a very large exoskeleton becomes a serious mechanical liability. The animal has to support its own weight against gravity, and the exoskeleton's mass scales faster than the muscles that move it, imposing a practical upper limit on size. In water, that constraint is relaxed. Buoyancy offsets a significant portion of the gravitational load, allowing the exoskeleton to be larger and heavier than would be viable on land. This is likely a significant part of why Eurypterids could reach sizes that no terrestrial arthropod has ever matched. The third major hypothesis is sometimes called the ecological vacuum argument, and it is perhaps the most intuitively compelling of the three. The idea is straightforward. In the Silurian and early Devonian, the large predator ecological niche in aquatic environments was not yet dominated by large vertebrates. Jawed fish were present, but had not yet produced the massive, heavily armoured apex predators that would emerge later in the Devonian. The placoderms, the armoured jawed fish that would eventually include giants like Jaekelopterus, were still relatively modest in size during the period when Jaekelopterus was at its largest. In the absence of large vertebrate competitors and predators, the ecological space for a large aquatic predator was open, and Eurypterids, already well established and diverse, were positioned to fill it. Without the competitive pressure that large fish would eventually bring, there was no strong evolutionary force pushing back against increasing body size. If larger individuals had advantages in capturing prey and avoiding what few threats existed, selection would have favoured growth, and the lineage could push toward the size limits imposed by physiology and mechanics rather than ecology. Each of these three explanations, the oxygen hypothesis, the buoyancy hypothesis, and the ecological vacuum hypothesis, captures something real about the conditions that made Paleozoic arthropod gigantism possible. And each of them has limitations. The oxygen argument does not account well for Jaekelopterus' time period. The buoyancy argument explains why aquatic arthropods could be larger than terrestrial ones, but does not fully explain why they were so much larger than their modern aquatic descendants. The ecological vacuum argument is compelling but somewhat circular, explaining size by the absence of competitors without fully accounting for the physiological mechanisms that allowed the size to be achieved in the first place. Most researchers today favour a combination of factors, with buoyancy and ecological opportunity probably playing larger roles for the Eurypterid specifically, and oxygen playing a more significant role for the later Carboniferous giants. What is perhaps most striking about this whole question is that it reminds us how contingent the living world is. The animals alive today are not the inevitable result of some predetermined trajectory. They are the survivors of a specific sequence of events, extinctions, climate shifts, evolutionary innovations, and ecological upheavals, each of which closed off some possibilities and opened others. The fact that no arthropod today comes close to the size of Jaekelopterus is not because arthropod body plans are fundamentally incapable of producing animals that large. The fossil record proves they can. It is because the specific combination of conditions that made Jaekelopterus possible, the shallow warm seas, the relative absence of large vertebrate predators, the mechanical support of water, and whatever other factors contributed, no longer exists in the same configuration. The largest arthropods alive today offer a useful point of comparison. The Japanese spider crab Macrochaera kempferi holds the record for the greatest leg span of any living arthropod, with legs that can stretch to 3.7 meters from tip to tip. But the body of a Japanese spider crab is relatively small, perhaps 40 centimeters across the carapace, and the animal's mass is modest compared to what its impressive leg span might suggest. The coconut crab, Burgus latro, is the largest living terrestrial arthropod by mass, reaching up to 4 kilograms in the largest recorded individuals, and it is a genuinely impressive animal, but it is operating near what seems to be a practical upper limit for land-based arthropods. Neither of these animals approaches the scale of Jaekelopterus, and both of them exist in a world where large vertebrates have long since established dominance over the largest predator niches. The numbers, then, tell a story that goes beyond any single animal. A 46 centimetre claw, a 2.5 metre body estimate, a size record that has held since 2007, a set of competing hypotheses that collectively explain the conditions for gigantism without any one of them fully closing the question. It is a picture built from fragments, in more than one sense of the word, and what it points toward is not just the remarkable biology of one extinct animal, but the deeper question of how the world that produced that animal eventually changed, what forces brought it to an end, and what the disappearance of Jaekelopterus and its kin tells us about the fragility of even the most dominant creatures in their time. That question leads us inevitably toward the extinctions, the slow decline of the Eurypterids, and the geological events that gradually dismantled the world Jaekelopterus had inhabited so completely. And so we arrive at a question that Jaekelopterus shares with other giants, a question that reaches beyond any single species and into the deeper patterns of life on Earth. Why did the ancient world produce creatures of such extraordinary size, and why does the modern world seem to have drawn back from those extremes? It is a question worth sitting with for a moment, because the answer is not a simple one, and the story it tells about our planet is genuinely remarkable. The phenomenon researchers sometimes call Paleozoic gigantism is not limited to Eurypterids. It shows up across multiple branches of the arthropod family tree, in different environments, at different times, and under different ecological conditions. Each case adds a piece to the picture, and together they suggest that the ancient world was not simply a scaled-up version of today, but a fundamentally different configuration of life, atmosphere, and opportunity. Perhaps the most famous example of ancient arthropod gigantism comes not from the sea but from the air. Meganeura, a dragonfly-like insect from the Carboniferous period, roughly 300 million years ago, had a wingspan estimated between 65 and 70 centimetres. To put that in terms that are a little easier to hold in your mind, that is roughly the wingspan of a modern red-tailed hawk. A dragonfly the size of a hawk. It is the kind of fact that sounds like something from a science fiction story, and yet it is simply what the fossil record shows. Meganeura was a predator, almost certainly capable of catching other flying insects and possibly small vertebrates, and it lived in the dense, humid forests of the Carboniferous in what is now Europe and North America. It was not alone. Related giant insects from the same period, grouped under the broader category of griffinflies, suggest that very large flying arthropods were a real and persistent feature of the Carboniferous world, not a rare accident. Then there is Arthroplura, which belongs to an entirely different branch of the arthropod tree and is perhaps the most visually striking of all the ancient giants. Arthroplura was a millipede, or more precisely a member of the extinct group Arthropluridae, and the largest known specimens reached an estimated length of around 2.5 metres. Some estimates push toward 2.6 metres for the largest fragmentary trackways attributed to this animal. It lived during the Carboniferous and early Permian, roughly 315 to 299 million years ago, and it appears to have been a plant eater, moving through the dense coal forests of its time. The trackways it left, fossilised impressions in ancient mud, are some of the most evocative trace fossils in the entire record. You can look at those parallel rows of imprints and feel, in a quiet way, the weight of something enormous moving through a forest that no longer exists. What connects Meganeura, Arthroplura, and Jaekelopterus is not just their size, but the fact that their size seems to belong to a specific window in Earth's history. A window that has since closed, and the question of why that window existed and why it closed is one of the more fascinating puzzles in paleontology. The hypothesis most people encounter first is the oxygen argument, and it is worth examining carefully because it is both compelling and more complicated than it first appears. The basic idea is that higher atmospheric oxygen levels would allow arthropods to grow larger by improving the efficiency of their respiratory systems. Insects and many other arthropods breathe through a network of tubes called tracheae, which carry oxygen directly to tissues without the intermediate step of a blood-based circulatory system. This system is highly efficient at small scales, but as body size increases, the tubes have to become longer and more branched, and at some point the diffusion of oxygen through them becomes a limiting factor. Higher atmospheric oxygen would extend the range over which this system could function effectively, potentially allowing larger body sizes. The Carboniferous period, when Meganeura and Arthroplura were alive, did indeed have elevated atmospheric oxygen. Estimates based on carbon and sulfur isotope ratios in ancient rocks suggest that Carboniferous oxygen levels may have reached somewhere around 30 to 35% of the atmosphere, compared to today's 21%. The Carboniferous was also the period of the great coal forests, vast expanses of tree-sized lycopsids and ferns that were producing and burying enormous quantities of organic carbon, which has the effect of elevating atmospheric oxygen as a byproduct. So the timing fits reasonably well for the Carboniferous giants, and the mechanism is at least plausible. But here is where the oxygen hypothesis runs into difficulty when we try to apply it to Jaekelopterus. Jaekelopterus lived during the Silurian and early Devonian, a period when atmospheric oxygen was not elevated in the same way. In fact, estimates for Devonian oxygen levels suggest they were somewhat lower than today, perhaps around 15 to 18% during parts of the period. If oxygen was the key driver of arthropod gigantism, Jaekelopterus should not have been able to reach 2.5 metres, and yet the fossil record says it did. This is the tension that makes the oxygen hypothesis insufficient on its own as an explanation for all Paleozoic gigantism. It may well explain the aerial giants of the Carboniferous, where the timing and the mechanism align, but it does not explain Jaekelopterus, which means we need other pieces. The buoyancy argument, which was touched on in thinking about Eurypterids specifically, offers a different kind of explanation, one that is grounded in the physics of living in water rather than the chemistry of the atmosphere. An aquatic animal does not have to support its full weight against gravity in the same way a land animal does. The water around it provides lift, distributing the mechanical load in a way that land cannot. For an arthropod, whose structural support comes from an external skeleton rather than an internal one, this matters enormously. The exoskeleton has to be moved, carried and maintained, and on land the energy cost of doing that scales quickly with size. In water, the cost is substantially reduced, and the practical upper limit on size is pushed considerably higher. This is why, if you look at the largest arthropods alive today, the ones with the most impressive dimensions are aquatic. The Japanese spider crab's remarkable leg span is possible partly because the animal spends its life in deep water, where its long, delicate limbs are supported by the medium around them. On land, those limbs would be mechanically unworkable. The coconut crab, which is genuinely terrestrial, is impressively large for a land arthropod, but it is still a fraction of the size of the ancient aquatic giants. The buoyancy argument helps explain why Eurypterids, living in water, could reach sizes that no land arthropod has ever matched, even during periods when atmospheric oxygen was not elevated. But the buoyancy argument also has its limits, as any honest accounting of the evidence has to acknowledge. Modern oceans contain enormous buoyant environments, and the largest arthropods living in them today are still nowhere near the scale of Jaekelopterus. If buoyancy alone was sufficient, we might expect to find giant Eurypterid-like animals in today's seas. We do not. Which means the buoyancy of the aquatic environment was a necessary condition for Eurypterid gigantism, but not a sufficient one. Something else also had to be in place. That something else is likely what researchers call the ecological opportunity argument, and it is perhaps the most straightforward of the three explanations in its logic. During the Silurian and early Devonian, the large predator niche in aquatic environments was genuinely open in a way it has not been since. The vertebrates that would eventually come to dominate those environments, the large armoured fish of the later Devonian, the sharks, the ray-finned fish, were either absent or still relatively modest in size and diversity. The seas were not empty, but the largest ecological role, the apex predator of a substantial body of water, was not yet claimed by anything with a backbone. Eurypterids were already diverse and well established. They had been present since the Ordovician, and had developed a wide range of body forms and ecological roles. When the opportunity to fill the apex predator niche was available, the Eurypterid lineage was positioned to take it. And in the absence of competitive pressure from large vertebrates, there was no strong evolutionary force acting against increasing body size. If larger individuals were better at catching prey and less vulnerable to the threats that existed, natural selection would have favoured growth, and the lineage could push toward the size limit set by physiology and mechanics rather than ecology. What makes this argument interesting is that it reframes the question slightly. Instead of asking what allowed Jaekelopterus to grow so large, it asks what prevented anything else from growing large enough to compete with it. The answer, in the early Devonian, is that nothing did. The vertebrate radiation that would eventually produce the dominant aquatic predators of later periods had not yet arrived in force. The window was open, and Jaekelopterus, along with its Eurypterid relatives, filled it. Now, it is worth pausing here to note something that these three explanations, the oxygen hypothesis, the buoyancy argument, and the ecological vacuum argument, share in common. None of them, taken alone, fully accounts for Paleozoic arthropod gigantism. Each one captures a real part of the picture, and each one has blind spots. The oxygen hypothesis fits the Carboniferous giants, but not Jaekelopterus. The buoyancy argument explains why aquatic arthropods could be larger than terrestrial ones, but does not explain why modern aquatic arthropods are so much smaller than their ancient counterparts. The ecological vacuum argument is compelling but somewhat circular, in that it explains large size by pointing to the absence of competitors without fully describing the physiological pathway by which the size was achieved. Most researchers who work on this question today favour a combination of factors, with different weights assigned depending on the specific animal and time period in question. For Jaekelopterus and the Eurypterids, buoyancy and ecological opportunity are probably the most important contributors. For the Carboniferous aerial giants like Meganeura, elevated atmospheric oxygen is a more plausible primary driver, though ecological factors likely played a role there too. For Arthroplura, the picture is less clear, and the debate continues. There is something genuinely satisfying about a scientific question that does not resolve neatly into a single answer. It is a reminder that the natural world is built from overlapping systems, and that the phenomena we observe are usually the product of multiple interacting causes rather than one clean mechanism. Paleozoic gigantism is one of those phenomena. It emerged from a specific configuration of atmosphere, ocean chemistry, evolutionary history, and ecological circumstance, and when that configuration changed, the giants it had supported disappeared. And this brings us to a quieter observation about what Jaekelopterus and its contemporaries represent. The living world is not a fixed state. It is a sequence of states, each one shaped by the conditions of its time, and each one eventually giving way to something different. The carboniferous coal forests that fed the oxygen spike and sheltered Meganeura are now the coal seams we mine. The shallow Devonian seas that Jaekelopterus patrolled are now the rock formations of Central Europe. The ecological vacuum that allowed Eurypterids to reach their greatest sizes was eventually filled, first by larger and more diverse fish, then by the broader vertebrate radiation that followed. What we see when we look at Paleozoic gigantism is not a world that was simply more extreme than ours, but a world that was differently configured and that produced different outcomes as a result. The giants of the Paleozoic were not aberrations or evolutionary experiments that failed. They were the logical products of the conditions that existed, filling the roles that were available, growing to the sizes that their environments permitted and their ecologies rewarded. The fact that those conditions no longer exist does not diminish what they produced. It simply places those animals in their proper context, as inhabitants of a world that has since been replaced by a different one. And that thought leads naturally toward the next part of this story, because the world that supported Jaekelopterus did not simply fade quietly. It ended in a series of catastrophic events that reshaped the oceans, rearranged the food webs, and began the long decline of the Eurypterids as a group. Understanding what happened to Jaekelopterus's world, and to the lineage it belonged to, means looking at some of the most dramatic geological events in Earth history. Events that left their mark not just on the Eurypterids, but on nearly everything alive at the time. The late Devonian is not an easy period to picture, because it requires holding two contradictory images at once. On one hand, it was a world of extraordinary biological richness, a time when fish were diversifying rapidly, when the first forests were beginning to take root on land, when the sea floor was thick with life, and the shallow coastal waters were among the most ecologically complex environments the planet had yet produced. On the other hand, it was a world moving toward catastrophe, and the catastrophe, when it came, was thorough enough that it fundamentally altered the trajectory of life on Earth. Jaekelopterus itself had likely already disappeared before the worst of what was coming. The genus is associated primarily with the early Devonian, with the fossil material from Prum dated to around 390 to 393 million years ago, which places it near the end of the early Devonian or the beginning of the Middle Devonian period. The great extinction events that would later devastate the oceans came later, toward the end of the Devonian, roughly 372 million years ago for the first pulse, and around 359 million years ago for the second. So when we talk about what ended Jaekelopterus's world, we are really talking about two separate but related things. There are the pressures that likely pushed Jaekelopterus itself toward extinction, which were probably competitive and ecological in nature, and then there is the broad catastrophe that eventually overtook the Eurypterids as a group, which was something far more violent and far-reaching. The pressures on Jaekelopterus during its own time were already significant. The Devonian is often called the Age of Fishes, and for good reason. It was during this period that jawed fish, the gnathostoms, underwent a dramatic radiation into an extraordinary variety of forms. Among the most important of these for our purposes were the placoderms, heavily armoured fish with bony head shields and powerful jaws, which became the dominant large predators of Devonian seas. Some placoderms grew to remarkable sizes. Dunkleosteus terrelli, perhaps the most famous of them, reached lengths estimated at around 4 to 6 metres, with a bite force that some researchers have estimated at over 4,000 newtons, making it one of the most powerful biters in the history of vertebrate life. Jaekelopterus and Jaekelopterus may have overlapped in time to some degree, though the precise temporal relationship between them is not fully established from the available fossil record. The arrival of large, fast, well-armed vertebrate predators in the same ecological space that Eurypterids had occupied would have created competitive pressure that the Eurypterids had not previously faced in that form. Fish have certain advantages over arthropods in an aquatic environment. Their endoskeleton allows for more efficient muscle attachment and greater speed. Their gills are highly effective at extracting oxygen from water. They do not need to molt their entire exoskeleton in order to grow, which means they do not have the same period of dangerous vulnerability that an arthropod faces during ecdysis. A fish can grow continuously and relatively safely in a way that a large arthropod simply cannot. And as the Devonian progressed, the fish were not just growing larger, they were also growing more diverse, filling ecological niches from the open water to the seafloor, from small prey-catching specialists to large apex predators. This does not mean that the rise of jawed fish directly caused the extinction of Jaekelopterus through simple head-to-head competition. Ecological replacement is rarely that clean or direct. What is more likely is that the increasing diversity and size of fish in Devonian seas gradually eroded the ecological advantages that Eurypterids had enjoyed during the Silurian and early Devonian, when vertebrate competition was more limited. As fish became more numerous, more varied, and more capable, the food resources available to large Eurypterids may have become harder to access, and the niche that Jaekelopterus occupied may have become less tenable. Over geological time, this kind of gradual competitive pressure can be as effective as any catastrophe. By the time the true catastrophes of the late Devonian arrived, Jaekelopterus was almost certainly already gone. But the Eurypterids as a broader group were still present, having survived as a lineage for over 100 million years by that point. And the late Devonian extinction events, known formally as the Kellwasser event around 372 million years ago, and the Hagenberg event around 359 million years ago, were among the most severe biological crises in the history of complex animal life. The Kellwasser event, named after a black shale layer found in the Harz Mountains of Germany, marks a horizon in the rock record where a vast number of marine species simply disappear. Roughly 70 to 80% of all invertebrate marine species are estimated to have been lost during the late Devonian extinction as a whole, with the Kellwasser event representing one of the most intense pulses of that loss. Reef ecosystems, which had been expanding and diversifying throughout the Devonian, collapsed almost entirely. The stromatoporoid sponges and tabulate corals that had built those reefs largely vanished. The trilobites, already in decline, were further reduced. Many of the placoderm fish that had dominated Devonian seas did not survive. The ocean chemistry shifted dramatically, with widespread evidence of anoxia, meaning low or absent oxygen levels in the water, particularly in deeper and mid-water zones. The causes of the late Devonian extinction remain a subject of active research and genuine scientific debate. Several mechanisms have been proposed, and most researchers today believe that multiple factors were acting simultaneously or in sequence rather than a single cause driving the entire event. One of the most discussed is ocean anoxia itself, the spread of oxygen-depleted water through large portions of the world's oceans. When oxygen levels in seawater drop below a certain threshold, most animal life cannot survive. The black shales associated with the Kellwasser event are themselves a product of anoxic conditions, formed when organic material accumulated on the sea floor without being broken down by oxygen-dependent decomposers. The spread of anoxic zones would have been devastating for any marine animal that required well-oxygenated water, and the evidence suggests that these zones expanded dramatically during the late Devonian. What caused the anoxia is itself debated. One hypothesis involves the expansion of land plants during the Devonian. As the first true forests spread across the continents during this period, their root systems began breaking down rock on a massive scale, releasing nutrients including phosphorus into rivers and eventually into the oceans. This nutrient influx would have fueled explosive growth of algae and other photosynthetic organisms in coastal waters, a process known as eutrophication. When those organisms died and sank, the decomposition process consumed enormous quantities of dissolved oxygen, creating the dead zones that the fossil record preserves. The idea that the greening of the land may have contributed to the death of the seas is one of the more quietly striking ironies in Earth's history. Volcanic activity has also been implicated, specifically the Vilui Traps, a large igneous province in what is now Siberia, which was active during the late Devonian. Large-scale volcanic eruptions can inject sulfur dioxide and carbon dioxide into the atmosphere, altering global temperatures, disrupting ocean chemistry, and triggering a cascade of environmental changes. The precise timing of the Vilui Traps activity relative to the extinction pulses is still being refined, but the correlation is considered significant by a number of researchers. There is also evidence of sea level changes during this period, with cycles of regression and transgression, meaning the oceans retreating and then advancing over continental shelves, which would have disrupted the shallow marine habitats that were home to the greatest concentration of life. Asteroid or bolide impacts have been proposed as contributing factors as well, and there is some evidence for impact events during the late Devonian, including the Alamo impact in what is now Nevada and the Woodley structure in Australia. Whether these were significant enough to drive extinction on their own is uncertain, and most researchers treat them as possible contributing factors rather than primary causes. The current picture of the late Devonian extinction is one of multiple stressors acting on ecosystems that were already under pressure, with the combination proving catastrophic in a way that no single factor might have been alone. The Eurypterids survived the late Devonian extinction, which is itself a remarkable fact given how thorough that event was. They continued into the Carboniferous and then into the Permian, though they were never again as diverse or as ecologically prominent as they had been during their Silurian and early Devonian peak. The fossil record shows a clear trend of declining diversity through the later Paleozoic, with fewer and fewer species represented in progressively younger rock formations. By the Permian, the Eurypterids were a shadow of what they had once been, a small remnant of a lineage that had once included over 250 known species and had produced the largest arthropod in Earth's history. What finally ended the Eurypterids was the Permian-Triassic extinction event, approximately 252 million years ago, the single largest mass extinction in the history of complex animal life on this planet. The numbers associated with this event are almost difficult to hold in the mind. Estimates suggest that somewhere between 90 and 96% of all marine species were lost, along with roughly 70% of terrestrial vertebrate species. The event was so thorough that it is sometimes referred to informally as the Great Dying, a name that captures something of the scale of what happened. The oceans did not simply lose species. In some regions, they appear to have lost the basic ecological structure that had supported complex communities for hundreds of millions of years. Recovery took millions of years, and the world that emerged on the other side of the Permian-Triassic boundary was profoundly different from the one that preceded it. The causes of the Permian-Triassic extinction are better understood than those of the late Devonian event, though significant questions remain. The primary driver is generally considered to be the Siberian Traps, one of the largest volcanic events in Earth's history, which erupted over a period of roughly 1 to 2 million years and released enormous quantities of carbon dioxide, sulfur dioxide, and other gases into the atmosphere. The resulting warming, ocean acidification, and widespread anoxia created conditions that the majority of marine organisms could not survive. The Eurypterids, already reduced to a small number of species by the time the Permian-Triassic boundary arrived, did not make it through. Their last known representatives appear in rocks from the late Permian, and after that, the fossil record is silent. There is something worth pausing on in that silence. The Eurypterids had existed for over 200 million years by the time they disappeared. They had survived the late Ordovician glaciation, the Silurian, the Devonian, and the Carboniferous. They had outlasted the placoderms that had once competed with them. They had persisted through multiple smaller extinction events and periods of environmental change. The lineage that produced Jaekelopterus was not fragile. It was one of the most enduring groups of complex animals in Earth's history, and yet the Permian-Triassic extinction, with its sustained volcanic forcing and cascading environmental effects, was something that even 200 million years of evolutionary resilience could not withstand. It is worth noting too what the world lost when the Eurypterids disappeared. Not just a group of animals, but a body plan, a set of ecological strategies, and a lineage of evolutionary experimentation that had been running for longer than the dinosaurs would later run. The Eurypterids had explored the possibilities of the chelicerate arthropod body form across an extraordinary range of sizes, environments, and lifestyles. Some had been tiny, under a centimetre. Some had been enormous. Some had been marine, some brackish, some possibly freshwater. Some had been active predators, some likely scavengers, some possibly adapted to partially terrestrial existence. When the last Eurypterid died sometime in the Permian, all of that diversity, all of that evolutionary history ended with it. There would be no descendants, no survivors, no lineage carrying those experiments forward. The horseshoe crabs, which are the closest living relatives of the Eurypterids, survived the Permian-Triassic extinction and persist today, but they are a separate branch of the chelicerate tree, not a continuation of the Eurypterid lineage itself. The extinction of Jaekelopterus, whenever exactly it occurred within the early to middle Devonian, was therefore not just the loss of one particularly large species. It was one early chapter in the long, slow decline of an entire order of animals, a decline that proceeded through competitive pressure, environmental change, and ultimately catastrophic extinction events across a span of more than 130 million years. The world that supported Jaekelopterus at its peak was already changing during its lifetime, and the changes would only accelerate. And yet, even in extinction, the Eurypterids left something behind. They left their fossils scattered through the rock record of multiple continents, preserved in the sediments of the seas they once inhabited. They left the evidence of their existence for later minds to find and interpret, including that single remarkable claw fragment from the Devonian rocks near Prum, the one that started this entire conversation. The Eurypterids are gone, but the record of what they were remains, waiting in stone, and that record tells us something important not just about those animals, but about the nature of life itself, about how body plans can be pushed to extraordinary extremes under the right conditions, and about what happens when those conditions change. Which brings us naturally to a question that the fossil record of Jaekelopterus raises in a very particular way. Because knowing that this animal existed, knowing roughly how large it was, knowing what it ate and where it lived and when it disappeared, is only part of the picture. The other part, the part that touches on something deeper about what evolution can produce and what physical constraints actually mean, is the question of how an animal this size actually functioned. What did it mean, mechanically and physiologically, to be an arthropod 2.5 meters long? And what does the existence of Jaekelopterus tell us about the true limits of the arthropod body plan, limits that modern animals suggest are far more conservative than the ancient record implies they needed to be? That question, the one about what it actually meant to be an arthropod of this size, is not purely a matter of curiosity. It touches on something fundamental about how life works, about the relationship between body design and physical reality, and about why the animals we see around us today look the way they do. Because the arthropod body plan, for all its extraordinary success across hundreds of millions of years, carries within it a set of constraints that become increasingly difficult to manage as size increases. And Jaekelopterus, at 2.5 meters, was pushing those constraints to a place that no arthropod before or since appears to have reached in quite the same way. The most fundamental of those constraints is the exoskeleton itself. Arthropods do not have internal skeletons the way vertebrates do. They wear their skeleton on the outside, a rigid external shell made primarily of chitin, a tough lightweight polymer that also forms the wings of insects and the shells of crabs. In most arthropods, this exoskeleton is reinforced with calcium carbonate or other minerals, giving it additional hardness. It is a remarkably effective structural solution for small and medium-sized animals, providing protection, a rigid anchor for muscle attachment, and a degree of waterproofing. But it comes with a cost that becomes more significant the larger the animal grows. To grow at all, an arthropod must shed its exoskeleton entirely, expand its soft body, and then wait for a new, larger exoskeleton to harden around it. This process is called ecdysis, and it is one of the most dangerous moments in the life of any arthropod. For a small crab or a juvenile eurypterid, ecdysis is already a vulnerable period. The animal is temporarily soft, slow, and exposed. Predators that could not pierce the hardened shell can now attack freely. The animal may struggle to move effectively while its new exoskeleton is forming. It must find shelter, avoid disturbance, and wait. For most small arthropods, this period lasts hours to days. But for an animal the size of Jaekelopterus, the implications of ecdysis are considerably more dramatic. A 2.5-metre eurypterid shedding its entire exoskeleton would have been, for however long the process took, one of the most physically vulnerable large animals in its ecosystem. The soft body would have been enormous, difficult to move quickly, and almost entirely unprotected. Whatever shelter existed in the shallow estuaries and lagoons Jaekelopterus inhabited, it would have needed to find it, and find it reliably, every time it grew. There is also the mechanical question of the exoskeleton's ability to support a body of this mass. On land, this would have been essentially impossible. The largest terrestrial arthropods alive today give us a useful reference point. The coconut crab, Burgus latro, is the largest land-dwelling arthropod currently living, and it reaches a body weight of roughly 3 to 4 kilograms, with a leg span of up to about 1 metre. It is already pushing the practical limits of what a land-based exoskeleton can support against gravity. The Japanese spider crab, Macrochira kempferi, achieves a leg span of up to 3.7 metres, making it the widest living arthropod, but its body mass is relatively modest, perhaps around 2 kilograms, because those long legs are thin and lightweight rather than heavily built. Neither of these animals is remotely close to what Jaekelopterus would have represented in terms of total body mass. The reason Jaekelopterus could exist at all, at least according to the most widely accepted explanation, is water. Buoyancy is the key. In an aquatic environment, the surrounding water supports a significant portion of the animal's weight, reducing the mechanical load on the exoskeleton and allowing structures that would collapse under their own weight on land to function perfectly well beneath the surface. This is why marine arthropods can grow larger than terrestrial ones, and it is one of the central reasons why the giant eurypterids, along with other large ancient arthropods, were overwhelmingly aquatic. The sea was not just where they lived, it was, in a very real sense, what made their size physically possible. But buoyancy alone does not resolve every physiological challenge that comes with being an arthropod of this scale. There is also the question of respiration. Eurypterids breathe through structures called book gills, thin, layered respiratory organs located on the underside of the abdomen, somewhat analogous in structure to the book lungs found in modern arachnids, though adapted for aquatic use rather than air breathing. Book gills work by passing water across thin, richly vascularized membranes, allowing oxygen to diffuse into the blood and carbon dioxide to diffuse out. In smaller eurypterids, this system was clearly adequate. But in an animal the size of Jaekelopterus, the question of whether book gills could supply enough oxygen to fuel a body of that mass is genuinely interesting, and not entirely resolved. The challenge is one of surface area relative to volume. As an animal grows larger, its volume increases much faster than its surface area. A body that is twice as long in every dimension has 8 times the volume but only 4 times the surface area. This means that larger animals need proportionally more efficient respiratory systems to supply oxygen to their tissues, or they need to have lower metabolic rates, or some combination of both. In vertebrates, this problem was solved through the evolution of increasingly complex internal respiratory structures, lungs and gills with enormous folded surface areas packed into relatively small spaces. Arthropods, working with book gills or tracheal systems, have more limited options for increasing respiratory surface area. One possibility is that Jaekelopterus had a relatively low metabolic rate, moving in slow, energy-efficient bursts rather than sustaining continuous high-energy activity. This would be consistent with the ambush predator model, suggested by its anatomy, lying in wait rather than pursuing prey across open water. Another possibility is that the book gills of Jaekelopterus were proportionally larger or more elaborately structured than those of smaller eurypterids. Though without a complete fossil specimen, this remains speculative. What is interesting about these physiological constraints is not simply that they existed, but what they imply about the conditions under which Jaekelopterus evolved. For an arthropod to reach 2.5 meters, every aspect of its biology had to be compatible with that size. The exoskeleton had to be strong enough to function, the respiratory system had to be adequate, the musculature had to be sufficient to move those large paddle limbs and operate those massive chelicerae, and the animal had to be able to complete ecdysis successfully throughout its life. The fact that Jaekelopterus apparently managed all of this, however imperfectly or precariously, is itself a remarkable piece of evidence about what the arthropod body plan is actually capable of under the right ecological and environmental conditions. And that is perhaps the most important thing that Jaekelopterus tells us about evolution and body plan limits. Not that arthropods can always be this large, but that under specific circumstances, with the right combination of environmental support, ecological opportunity, and evolutionary time, the arthropod body plan can be pushed far beyond what we might predict from looking at the animals alive today. Modern arthropods are constrained not only by physics but by ecology. They exist in a world dominated by vertebrates, in which large predatory niches are occupied by fish, reptiles, birds and mammals. The ecological space that Jaekelopterus occupied simply does not exist anymore, not because arthropods became incapable of filling it, but because other animals got there first and have held it ever since. This connects to a broader concept in evolutionary biology sometimes called convergent evolution, though what Jaekelopterus illustrates is perhaps more specifically the idea of ecological opportunity shaping body plan expression. Convergent evolution typically refers to unrelated lineages independently evolving similar features in response to similar selective pressures. What we see in the giant eurypterids is something slightly different, a single lineage exploring the extreme end of its body plan's potential because the ecological conditions of the time made that exploration not just possible, but advantageous. The shallow, warm, prey-rich waters of the Silurian and Devonian, combined with the relative absence of large vertebrate predators, created a context in which growing larger conferred real advantages, and the eurypterid body plan, flexible and adaptable as it was, responded accordingly. It is worth pausing on what this means for how we think about the limits of biological design. There is a tendency, perhaps natural and understandable, to look at the animals alive today and treat them as representing the range of what is possible. We see the largest living arthropods and assume they mark some kind of ceiling. But the fossil record consistently challenges that assumption. It shows us that the range of what is biologically possible is not fixed. It shifts with atmospheric composition, with ocean chemistry, with the presence or absence of competing lineages, with the structure of ecosystems that have no modern equivalent. Jaekelopterus is one of the clearest demonstrations of this principle that paleontology has yet produced. A single animal that proves, simply by having existed, that the arthropod body plan can reach scales that nothing alive today comes close to matching. There is also something worth noting about what Jaekelopterus does not tell us. which is equally important in its own way. The existence of one giant species does not mean that gigantism was the norm for Eurypterids, or that the conditions of the Devonian routinely produced animals of this size. The vast majority of Eurypterid species were small to medium-sized, and even among the largest genera, Jaekelopterus stands out as exceptional. Pterygotus anglicus, one of the other large Eurypterids, reached an estimated 1.75 metres. Acuteramus, another large genus, is estimated at around 2 metres. These are impressive animals, but they are meaningfully smaller than Jaekelopterus. The 2.5 metre estimate for Jaekelopterus places it in a category of its own, not just the largest Eurypterid, but the largest arthropod we currently have evidence for in the entire fossil record. That distinction matters because it means Jaekelopterus was not simply a slightly larger version of something common. It was an outlier, an extreme expression of what the Eurypterid lineage could produce. And outliers in evolution are often the most informative data points we have, because they mark the edges of what a body plan can achieve, the places where biology strains against physical and physiological limits, and either finds a way through or fails. Jaekelopterus, as far as we can tell, found a way through, at least for a time. The molting question is worth returning to one more time, because it sits at the intersection of all these considerations in a way that is difficult to fully resolve, but impossible to ignore. Every time Jaekelopterus grew, it had to shed an exoskeleton that, by the time the animal was fully grown, would have been a structure of considerable size and complexity. The chelicerae alone, at 46 centimetres each, would have required careful extraction during the molting process. The paddle limbs, the walking legs, the broad prosoma shield, the long segmented abdomen, all of it would have needed to be withdrawn from the old shell and expanded into a new one. The logistics of this for an animal of this size in an aquatic environment with its own physical dynamics represent a genuine biological puzzle. We do not know how Jaekelopterus managed it. We do not know how long the process took, where the animal went during its vulnerable period, or what mortality rate was associated with molting at this scale. These are questions that a complete fossil specimen might begin to answer, though even then soft tissue processes leave limited traces in stone. What we can say is that Jaekelopterus solved these problems well enough to exist, well enough to grow to its recorded size, and well enough to persist as a species for whatever portion of the Devonian it occupied. The solutions it found are lost to us, buried in the details of a biology we can only partially reconstruct. But the fact that solutions were found at all is the point. It is the evidence that the constraints we identify when looking at modern arthropods are not absolute walls. They are boundaries that, under the right conditions, can be moved. And that is a thought worth sitting with for a moment, because it applies not just to Eurypterids, but to the broader story of what life on this planet has been, and what it might yet be. The history of life is full of body plans being pushed to extremes that later seem impossible, then becoming impossible again when conditions change, then being achieved again in different lineages under different circumstances. Jaekelopterus is one chapter in that story, a particularly vivid one, but one that connects to a much larger pattern of life testing its own limits across deep time. The fossil record of Jaekelopterus, fragmentary as it is, therefore carries more information than its size alone suggests. It tells us about the physics of aquatic exoskeletons, about the respiratory limits of bookgill systems, about the ecology of Devonian estuaries, about the evolutionary dynamics of a lineage under conditions of low vertebrate competition, and about the fundamental flexibility of biological design when ecological opportunity opens a door wide enough. All of that from a single claw fragment, 46 centimetres long, pulled from a rock formation in the Rhineland-Palatinate region of Germany, and described in a paper published in the journal Biological Letters in 2007. It is a remarkable amount of information to extract from one piece of ancient chitin, and yet, as informative as that fragment has been, it also represents the limit of what we currently know with any confidence. Because the questions that remain about Jaekelopterus, the ones that cannot be answered from a single claw, no matter how carefully it is studied, are in some ways even more interesting than the ones that have already been addressed. And those unanswered questions lead us directly into the territory of what the fossil record as a whole can and cannot tell us about an animal like this, and what it would take to know more. That question, what it would take to know more, is one that paleontologists who work on Eurypterids carry with them quietly, not as frustration exactly, but as a kind of ongoing awareness that sits just beneath every paper published and every reconstruction drawn. Because the honest answer is that what we currently have of Jaekelopterus rhenanii is, by any reasonable measure, very little. A single cheliceraal fragment, 46 centimetres of ancient chitin, preserved well enough to measure, well enough to compare, well enough to build an estimate from, but not nearly enough to answer the deeper questions about what this animal actually was in full, living detail. To understand why this matters, it helps to think about what a complete fossil would actually reveal, and how different that picture would be from the one we currently hold. A complete specimen, the kind that occasionally emerges from exceptionally preserved sites, would show us the internal arrangement of the body segments in precise detail. It would show us the exact proportions of the paddle limbs relative to the walking limbs, which would clarify how Jaekelopterus moved through the water and whether it was primarily a swimmer or a bottom dweller that used its paddles only occasionally. It would show us the gill structures, which are critical for understanding how the animal breathed and whether its respiratory system could realistically support a body of 2.5 meters. It would potentially preserve traces of the digestive tract, the nervous system layout, and in rare cases, even reproductive structures. Growth stages preserved in a single population sample would tell us how fast Jaekelopterus grew, whether it reached its maximum size quickly or slowly, and what a juvenile of this species looked like. None of that information is available to us right now. We have the claw, and from the claw, a careful estimate. Everything else is inference. The problem is not that scientists have been careless or that the claw has been poorly studied. The problem is taphonomic, which is the scientific way of saying it is a problem rooted in how organisms become fossils in the first place, and why so many of them do not. Taphonomy is the study of what happens to an organism between the moment it dies and the moment it is found, and it is a field that does a great deal to explain why the fossil record looks the way it does, which is to say, incomplete in very specific and often frustrating ways. Large animals with lightly mineralized bodies are among the hardest organisms to fossilize well. They tend to decompose quickly after death. Their soft tissues vanish within days or weeks in most environments. Their exoskeletons, if not buried rapidly in fine-grained sediment with low oxygen levels, will break apart, scatter, and dissolve. The larger the animal, the more surface area is exposed to decomposition, and the less likely it is that any significant portion of the body will survive long enough to be buried in a condition that allows fossilization. Jaekelopterus, at 2.5 meters, would have been a large and complex structure to preserve. Even the exoskeleton, which is more durable than soft tissue, is made of chitin rather than the dense mineral structures of bone or shell, and chitin degrades over geological time unless conditions are exactly right. The fragment that was found near Prüm in the Rhineland-Palatinate region survived because it happened to be buried in the right kind of sediment, in the right kind of chemical environment, at the right time. That is not a small set of requirements. It is a narrow window of circumstance, and the fact that even this single fragment made it through 400 million years to be found and described is, in its own way, a minor geological miracle. This is where the concept of Lagerstätten becomes relevant, because these exceptional preservation sites are the places where paleontology gets its clearest windows into ancient life, and their absence from the Eurypterid record is one of the reasons our picture of Jaekelopterus remains so partial. Lagerstätten, a German word meaning roughly storage place or motherlode in geological contexts, are sites where conditions were so precisely right that organisms were preserved with extraordinary completeness and detail. The Burgess Shale in British Columbia, formed around 508 million years ago, has yielded soft-bodied animals preserved in such detail that individual gill filaments and digestive traces are visible. The Solnhofen limestone in Bavaria, from the late Jurassic roughly 150 million years ago, preserved Archaeopteryx with feather impressions intact, along with jellyfish, insects, and delicate marine creatures that would otherwise leave no trace at all. The Messel Pit in Germany, from the Eocene around 47 million years ago, has produced mammals with fur, stomach contents, and even the iridescent colour of insect wings. No Lagerstätte has yet yielded a complete Eurypterid of the Jaekelopterus scale. There are sites where Eurypterids have been found in reasonable condition. The Silurian deposits of New York State have produced some of the best preserved Eurypterid material known, and various European and North American sites have contributed important specimens from different species. But the combination of the right preservation conditions and the right location, where a giant Eurypterid happened to die and be buried, has not yet produced the kind of specimen that would transform our understanding of Jaekelopterus from an estimate to a confirmed picture. It is possible that such a specimen exists somewhere in rock that has not yet been excavated, or has not yet been exposed by erosion, or lies beneath ground that no one has thought to look at carefully. Paleontology is, among other things, a science of finding things in places no one expected, and the history of the field is full of extraordinary specimens turning up in formations that had been studied for decades without yielding anything remarkable. There are also hints, less formally documented than the Prumchelicera, that other large Eurypterid material may exist in collections or in the literature in forms that have not yet been fully described. Paleontological collections at museums and universities around the world contain material that has been catalogued but not yet studied in depth. Fragments and partial specimens that may represent significant finds waiting for the right researcher to take a close look. It would not be surprising if some of this material, when examined carefully, turned out to belong to giant Eurypterids, possibly even to Jaekelopterus itself, or to closely related species of comparable size. The field moves slowly by necessity, because the work is painstaking, and the expertise required is specialised, and there is always more material than there are researchers with the time and resources to study it thoroughly. Beyond the question of what remains to be found in existing collections, there is the broader and more unsettling possibility that even larger Eurypterids existed and left no trace at all. This is not a fringe idea. It is a straightforward implication of what we know about taphonomy and about the incompleteness of the fossil record. The animals that get preserved are not a representative sample of the animals that lived. They are a biased sample, skewed toward organisms that lived in environments where fossilization was likely, that had hard parts that could survive, and that happened to die in places where burial occurred quickly. Animals that lived in open water, far from sediment-rich environments, would rarely be preserved. Animals that died in high-energy environments where currents and scavengers dispersed their remains would leave little behind. Animals that were simply rare, even if they existed for millions of years, might produce only a handful of fossils across their entire range and duration, and if those fossils happened to be deposited in rock that was later subducted, metamorphosed, or eroded away entirely, they would leave no record at all. The Devonian period lasted roughly 60 million years. Jaekelopterus, or something like it, may have existed for a significant portion of that time. Over 60 million years across the shallow seas and estuaries of what is now Europe, North America and other regions, there would have been many individual animals, many generations, many deaths. And yet we have one claw fragment. That ratio, one fragment to 60 million years of potential existence, is a stark reminder of how thin the fossil record actually is, even for animals that were large and presumably common enough to sustain a breeding population across geological time. If something larger than Jaekelopterus existed, something that lived in slightly different environments, or died in places where preservation was even less likely, we might have nothing. Not a fragment, not a trace, nothing at all. The absence of evidence is not evidence of absence, as the saying goes, And in paleontology, this is not just a philosophical comfort. It is a working principle that shapes how researchers interpret gaps in the record. What this means, practically, is that the 2.5 metre estimate for Jaekelopterus represents a known lower bound for Eurypterid size, not an established maximum. The largest Eurypterid we have evidence for was at least this big. Whether anything larger existed is a question the fossil record, as it currently stands, cannot answer. It is a genuinely open question, and it sits alongside many other open questions about Jaekelopterus that the single cheliceraal fragment cannot resolve on its own. There is something worth pausing on here, in the spirit of honesty about what science is and how it works. It can be tempting when reading about ancient animals to treat the reconstructions and estimates as if they were as solid as a photograph, as if the image of a 2.5 metre sea scorpion moving through a Devonian estuary is something we know rather than something we have carefully inferred. The distinction matters. The inference is well founded. It is based on rigorous comparative anatomy, on peer-reviewed methodology, on the work of researchers who understand Eurypterid biology as deeply as anyone alive today. The estimate of 2.5 metres for Jaekelopterus is not a guess in the casual sense. It is a scientific estimate, which means it is the best current answer to the question, supported by the available evidence, and open to revision if better evidence emerges. But it is still an estimate. The complete animal has never been seen. Its true proportions, its exact weight, its precise appearance, its behavioural repertoire, its internal anatomy, all of these remain in the territory of informed inference rather than confirmed fact. And that gap between inference and confirmation is not a failure of the science. It is simply the honest condition of working with a fossil record that preserves only fragments of the past, and working with them as carefully and rigorously as possible, while remaining clear about what is known and what is reconstructed. This is, in a way, one of the most quietly remarkable things about paleontology as a discipline. It is a science that operates at the edge of what can be known, that extracts extraordinary amounts of information from extraordinarily limited evidence, and that does so with a kind of patient, methodical care that rarely gets the attention it deserves. The researchers who studied the Prum chelicera, who measured it, who compared it to related species, who ran the scaling calculations and published their findings in biological letters in 2007, were doing something genuinely difficult. They were looking at a single piece of ancient chitin and asking what it could tell them about an animal that no human being has ever seen, that lived in a world that no human being has ever inhabited, that moved through ecosystems that no longer exist anywhere on Earth. And they answered that question as well as the evidence allowed, which is all that science can ever do. The incompleteness of the Jaekelopterus record is therefore not just a frustration, it is also an invitation. Every field season in the Devonian formations of Germany, the United Kingdom, North America and elsewhere carries the possibility of a find that would change what we know. A more complete specimen, even a partial one with multiple body segments preserved, would be transformative. It would allow researchers to test the scaling estimates directly, to confirm or revise the 2.5 metre figure, to examine the gill structures and the limb proportions and the details of the prosoma that are currently only reconstructed from related species. It would be, for anyone working on Eurypterid paleontology, the kind of discovery that reorients a field. And that possibility that somewhere in the Devonian rock of the Rhineland or the Scottish borders or the sedimentary formations of upstate New York, there might be a slab of stone containing something more complete than what has been found so far, is one of the things that keeps the questions about Jaekelopterus alive and interesting. It is not a closed case. It is an ongoing investigation, one that has been running since 2007, and that could be advanced at any moment by the right exposure of rock in the right place at the right time. That is the nature of paleontology, and it is one of the things that makes it a field of genuine and continuing excitement, even when the pace of discovery is slow and the evidence is fragmentary. What we know about Jaekelopterus as it stands is enough to place it firmly in the record as the largest arthropod yet discovered. What we do not know about it is enough to keep researchers working, keep questions open, and keep the possibility alive that the picture we have now is only a beginning. And that combination, a well-supported foundation of evidence and a wide-open horizon of unanswered questions, is perhaps the most honest and most compelling state that any scientific subject can be in. Not settled, not speculative, but genuinely in progress. The claw in the German rock formation started a conversation about size, about biology, about the ancient world that has not yet finished. And the next chapter of that conversation may be waiting in a hillside somewhere, in a layer of Devonian sediment that has not yet been exposed to daylight, where another fragment of this extraordinary animal is still, slowly, becoming something a future researcher will one day find and describe. That thought, quiet as it is, carries its own particular kind of wonder, and it connects naturally to the larger question of what Jaekelopterus ultimately leaves behind, not just in rock, but in our understanding of what life on this planet has been, and what it might still surprise us with. And so we return, as perhaps we always knew we would, to that shallow sea. Picture it one more time. Not the Germany of today, with its forests and its roads and its old stone towns, but the Germany of 400 million years ago, which was not Germany at all, not in any sense we would recognise. It was a warm, low-lying coastal margin sitting close to the ancient equator, covered in brackish water that was neither fully ocean nor fully river, but something in between. The sky above it was a different shade of blue, filtered through an atmosphere with a slightly different chemistry. There were no birds crossing that sky, no calls, no wing beats, nothing warm-blooded moving anywhere near the surface of the water. Only the slow drift of early plant life on the shores, the soft movement of algal mats on the sediment below, and the quiet, enormous presence of animals that would leave almost no trace of themselves behind. Into that water, something very large moved. It moved the way large things move in water, with a kind of unhurried authority, displacing the current around it rather than fighting through it. Its paddle limbs swept in slow, measured strokes. Its compound eyes, mounted flat on the top of its broad head shield, caught the light filtering down from the surface. Its chelicerae, each one nearly 1/2 metre long, held loosely at rest as it moved, ready to close when the moment required it. Everything about it was built for this world, for this water, for this particular slice of deep time that no living creature will ever experience again. And then, at some point in the Devonian period, it was gone. Not dramatically, not in a single catastrophic moment, but gradually, the way most things end in the long arc of evolutionary history. The world changed around it. The seas shifted. The fish grew larger and more capable. The ecological conditions that had allowed something of this scale to exist slowly ceased to be what they were. Jaekelopterus did not survive to see the end Devonian extinctions that would reshape so much of the marine world. It had already passed from the record by then, leaving behind only the faintest signature of its existence. A single claw, 46 centimetres of fossilised chitin, preserved in a slab of Devonian rock near Prüm in the Rhineland-Palatinate region of Germany. Found, eventually, by people who knew what they were looking at, measured, compared, published, and placed into the scientific record as the largest arthropod fragment ever discovered. That is all that remains of an animal that was almost certainly longer than a tall man is tall, that may have weighed as much as a large motorcycle, that moved through its world as one of the most formidable predators its ecosystem had ever produced. There is something almost vertiginous about that reduction. From a living animal, enormous and real, moving through warm water in a world completely unlike our own, to a single piece of fossilised claw in a rock formation. The distance between those two things, the living creature and the fragment, is not just physical, it is temporal, almost incomprehensibly so. 400 million years is a number that resists genuine understanding. It is long enough that the continents have rearranged themselves, long enough that the very chemistry of the atmosphere has changed multiple times, long enough that the lineages of animals that shared the water with Jaekelopterus have, in many cases, transformed beyond recognition or vanished entirely. And yet the claw remained, pressed into the sediment, slowly becoming stone, waiting in the dark of the rock for a moment that would not come for 400 million years. When Marcus Poschmann and his colleagues examined that fragment and published their findings in Biological Letters in 2007, they were doing something that deserves a moment of quiet appreciation. They were reaching back across that entire span of time and recovering something real from it. Not imagined, not invented, not extrapolated from mythology or story, but genuinely recovered from the physical record of the planet itself. The rock had preserved it. The science had read it. And from that single preserved piece, an entire animal, its scale, its probable anatomy, its ecological role, its place in the history of life, became something we could begin to understand. That is what one fragment can do when it is the right fragment, studied by the right people, in the right context. It can open a window into a world that would otherwise be entirely invisible to us. This is one of the things that makes deep time so extraordinary to think about quietly, at the end of a long day, when the mind is ready to slow down and let something genuinely large move through it. We tend to think of the past as something that recedes, that becomes less real the further back it goes, that fades into abstraction once it is old enough. And there is a sense in which that is true. We cannot visit the Devonian. We cannot stand on the shore of that ancient sea and watch the water. We cannot hear whatever sounds that world made, which were probably not many, at least not the kinds we would recognise. In almost every practical sense, that world is gone. And yet it left evidence. Everywhere, in the rock beneath our feet, in the sedimentary formations that geologists read like a slow and patient text, the history of life on this planet is preserved in fragments. Not completely, not evenly, not without enormous gaps and silences, but genuinely physically preserved. The claw of Jaekelopterus is not a metaphor. It is not a symbol. It is an actual piece of an actual animal that lived in an actual sea, and it is sitting in a museum collection right now, measurable and real, available to anyone with the training and the patience to study it. That fact, simple as it sounds, carries a kind of weight that is worth sitting with for a moment. The world is older than we can truly feel, and it has been recording itself all along. What Jaekelopterus leaves behind, beyond the physical fragment, is something harder to name but no less real. It leaves behind an expansion of what we understand to be possible. Before that Chelicea was found and measured, the upper limit of arthropod size in the fossil record sat somewhat lower than it does now. The discovery pushed the boundary outward. It demonstrated concretely and with good evidence that arthropod body plans are capable of reaching scales that dwarf anything alive today. That the largest crab, the largest lobster, the largest spider alive in the modern world are not approaching the ceiling of what arthropod biology can produce. They are simply what arthropod biology produces now, under current conditions, in the current world. Under different conditions, in a different world, the ceiling was considerably higher. That is a significant thing to understand, not just for paleontology, but for the broader question of how life works. Evolution is not a march toward any particular form. It is a response to conditions. When conditions allowed giant arthropods existed. When conditions changed, they did not. The animals that fill the large predator niches today are vertebrates, fish and marine mammals and large reptiles, because vertebrate body plans turned out to be more scalable under the conditions that followed the Devonian. But for a long stretch of deep time, before vertebrates had developed the size and capability to compete at the apex of aquatic food webs, arthropods held that position, and they held it at scales that we are only now beginning to fully appreciate. There is also something quietly humbling in what Jaekelopterus tells us about the incompleteness of our picture. If a creature of 2.5 meters, the largest arthropod in the fossil record, is known only from a single claw fragment, then what else might be out there, known only from an even smaller piece, or not yet known at all? The fossil record is not a complete archive. It is a sparse and uneven sampling of the history of life, biased toward animals with hard parts, biased toward environments where preservation is possible, biased toward the places where people have looked and the rock has been exposed. There are almost certainly animals that lived in the Devonian, some of them large, some of them ecologically important, that left no trace whatsoever. Not because they did not exist, but because the conditions for their preservation were never met. That possibility, that the Devonian Sea contained giants we have not yet found and may never find, is not a source of frustration so much as a reminder of scale. The history of life on this planet is larger than the fossil record, which is itself larger than what we have so far discovered, which is itself larger than what any single researcher or team or generation of scientists can fully process. We are always working with a partial picture, and the partial picture we have, including Jaekelopterus at its centre, is already extraordinary. There may be more out there. That is worth saying plainly and without drama, because it is simply true. The Devonian formations of Europe and North America have not been exhausted. New exposures happen every year, through erosion, through construction, through deliberate geological survey. Every one of those exposures is a potential window into a part of the record that has not yet been read. A more complete Jaekelopterus specimen, if one exists somewhere in the rock, would be one of the most significant paleontological finds of the century. It would resolve questions about body proportion, about gill structure, about the exact configuration of the limbs that are currently only answered by inference from related species. It would give us a far clearer picture of what this animal actually was. And beyond Jaekelopterus specifically, there may be other giants waiting, other eurypterids that reached comparable or greater sizes, preserved in formations that have not yet yielded them. Other arthropods from other periods and other environments whose scale we have not yet imagined because we have not yet found the evidence. The fossil record has surprised us before, repeatedly and dramatically, and there is no reason to think it has stopped doing so. So the story of Jaekelopterus is not, in the end, a closed story. It is a story that began with a discovery in 2007, and that is still being written slowly, by researchers working in the field and in the laboratory, by the slow erosion of Devonian rock in the Rhineland and elsewhere, by the patient accumulation of evidence that is the actual texture of scientific progress. The animal lived, was extraordinary, vanished, and left behind a fragment that opened a conversation about deep time and biological possibility that has not yet finished. That conversation is one of the most interesting ones science is having, even if it happens slowly and quietly and without much fanfare. It is a conversation about what life is capable of, about how far body plans can be pushed under the right conditions, about how much of the history of this planet remains to be recovered from the rock and understood. And Jaekelopterus, the largest arthropod we have ever found, the creature that was bigger than a human being and moved through a sea that no longer exists, sits at the centre of that conversation as a piece of evidence that is both remarkable in itself and suggestive of how much more there might be. There is a particular kind of wonder that comes from thinking about deep time not as a void, not as an absence of the familiar, but as a full and inhabited world, different from ours in almost every detail, but real in exactly the same way ours is real. The Devonian Sea was not a backdrop. It was a living system, complete and complex, full of animals doing what animals do, eating and moving and competing and dying, across millions of years and across geographies that no longer exist in any recognizable form. Jaekelopterus was part of that system. It was not a monster. It was not an anomaly. It was an animal, shaped by its environment, filling a role in its ecosystem, living out its span of time in a world that suited it. The fact that we can know this, even incompletely, even from a single claw, feels like something worth pausing to appreciate. The rock remembered, the science listened, and now, on a quiet night, somewhere far removed from the Devonian in every possible sense, you have spent a little time thinking about a creature that most people will never hear of, that lived and died 400 million years ago, and that left behind just enough of itself to be found. That is, in its own quiet way, a kind of miracle. Thank you for spending this time here, in this particular corner of deep time and careful science. If this journey through the ancient world brought you something, a little wonder, a little calm, a little of that specific pleasure that comes from learning something genuinely surprising about the history of life, then please do consider leaving a like, and if you have not yet subscribed to Life Beyond Seen, this would be a lovely moment to do so. There are many more stories like this one waiting here. Stories of lost worlds and strange animals and the patient, extraordinary work of understanding how the planet we live on came to be what it is. Come back whenever the night feels right for that kind of thinking. Sleep well, wherever you are, and carry with you, if you like, the quiet image of something enormous and real, moving through warm ancient water in a world that was already old when it was new.